And greetings, everybody. Welcome to the Evidence and Reasons for the Christian Faith video channel, home of YouTube's #1 creationist, as voted by fellow YouTubers. This presentation is probably going to be a little clunky tonight. I was dealing with some emergencies at the church. Some of you may know that the church I'm involved with, I'm a member of, I had a special indulgence from the elders to be a dual member at Occoquan Bible Church in McLean Bible. Occoquan is a small church, maybe less than 200 members. McLean had attendance of like 19,000 at its zenith. They meet in a $150 million complex, so we're talking big money here. If you're wondering how a church could be that wealthy, we had some very affluent patrons, including internet mogul who was a Christian, and praise God that there were people in the tech industry 20 years ago that loved McLean, and God blessed their business, and McLean was able to get this enormous property that's probably now valued anywhere from 100 to 200 million. So I'll put in $150 million there. They're in the middle of a business struggle within the church. If the church is dissolved, we don't know who might buy it up. And there could be a lot of people that could make a lot of money in the process if the church is dissolved. And so there are competing interests. And not to mention, fundamentally, there's also really disagreements about what's being taught. There has been allegations of a social justice, woke gospel being preached at McLean Bible. And there have been now very visible, nationally known fights now that are at McLean. It's all over the church headlines and church newspapers. And I didn't want to get involved in the fight, but I'm in the thick of things now. I will be broadcasting a lot of viewpoints from hopefully both sides. I'm afraid there's one side, which is the current church leadership, that has been so far reluctant to make appearances publicly and give an account for the decision making there. But I will seek to be part of inviting people. Yes, let the cash stabbing begin. Give an account. I mean, when you have a $150 million complex and a pulpit, you could speak from such a complex. This is a lot of political power. Donald Trump and Mike Pence went to that church. Okay, Donald Trump, Mike Pence, a lot of dignitaries, the King of Tongo, and senators, congressmen, because McLean Bible is right there near D.C. and not too far away from the White House. And we believe that God has placed that very conservative church, up until recently conservative, for a reason. And the conservatives are fighting what they believe is a left-wing takeover, a left-wing Marxist takeover of the church. And if you've been hearing a lot about the Southern Baptist Convention and their president, who's a plagiarist and woke and apparently having some ethical issues, big time, McLean's affiliation with them, which is in violation of McLean's own constitution, is raising issues. We don't know how much money flow is being pushed around, and this is disturbing. There's not been financial transparency. So I'm just saying the stakes are really big, and even though my little dinky contributions in terms of money to the church represents only the contributions, the cash contributions of one individual, I'm concerned at the stewardship of how this is going. Now, one way to avoid all this mess is maybe not to have mega churches in the first place and such large amounts of joint property that are, I mean, I could honestly foresee if the church continued to grow, if they had another complex, this could be like a $500 million enterprise. I mean, this is big time. And when that amount of money is being pushed around, you can get some interesting conflicts that come up. I do not know. The congregation, for example, doesn't know what the net total income of these pastors are. We want them to give accounting for the revenue flow from McLean Bible directly, and then their indirect sources. If they're tied in with the SBC and the SBC is sending them money, that needs to be disclosed. What sort of numbers are we talking about for pastor salaries? How about $250,000? That's a number I've heard floating around. One way to quash the rumors is to have transparency. We don't have that. Do you feel comfortable with a pastor earning $250,000 and talking about white privilege? I'm sorry. And I want to be fair. Give the other side a chance to come clean. And I've raised those concerns. I mean, I hope I'm wrong. God bless them if they're dirt poor and they're earning money that's below the poverty line. That'd be just incredible. I mean, I've always loved pastors that were bivocational. These guys had an independent living, and at Occoquan Bible, we had a lot of the pastors there. 2/3 of the pastors had an independent living. Air Force colonel, 2 of them are attorneys. Very blessed. One of them was vice president of USA Today. Now that's an interesting story because he became a conservative Christian sometime after he rose to that height, and you can imagine that caused him a lot of problems. But it was just glorious to have these pastors that were independently supported by means. So that's a little bit of side chit chat. I usually try to do that so that give people an opportunity for YouTube to notify them and join. So the topic tonight, so that's kind of a big apology what's going on in my world, and I want to give glory and honor to God. He is, He is supplying all my needs, and I've been blessed. And there are always challenges, but I want to try to do a good job for you all tonight. We're going to have a real nerd session tonight. Okay, so you've seen me kind of going easy and trying not to talk about too many technical topics. We've talked even about silly things, but tonight will be seriously technical, and so you can pray for me that this goes well, because I slapped this together with minimal time. So the first bit of news is Creationist score in peer review. Now technically these are ID proponents, and one of them, namely Ola Hossjer, I don't know if he's made any public statements about his views on intelligent design or creationism, and I'm going to respect that. He's actually a professor of mathematics and population genetics, tenured professor, published, nationally recognized. He got an award in Sweden for Mathematician of the Year. That's not easy to do. And he joined Gunter Bechly, who is that paleontologist who was a Darwinist. He organized the big Darwin celebration, and then when he began reading ID material, that he had a pile of books that he was featuring, trying to kind of mock the ID movement. He decided to read some of the books he was mocking, and he changed his mind. He is a professing Christian, perhaps a little unorthodox. I don't know. It's nuanced. Okay, so I don't know even what church. I think he goes to a Catholic church, so maybe depending on your definition of orthodox, that may be orthodox to you. But he accepts intelligent design. To my understanding, he accepts universal common ancestry, like a lot of ID proponents. But he does call himself, he has on and off called himself some sort of creationist. If I'm misquoting him, please forgive me, Gunter. No ill intent. People also know about Ann Gauger. She is a MIT, University of Washington, Harvard-trained biologist, molecular biologist, very intelligent, works for the Discovery Institute. So we have this paper that came out. We have a population geneticist, award-winning mathematician, and Ola Hossjer, professor. We have Gunter Bechly, a paleontologist. At the time, he was celebrated up until he became an ID proponent, until he became a Christian and then an ID proponent. And then we have Ann Gauger, who is a Harvard-trained, MIT-trained, University of Washington-trained biologist. Stellar qualifications, and certainly stellar relative to people like Dave Farina. Sorry to pick on Dave, but he's kind of our punching bag. So let me just read the abstract. It's gonna give me a headache to read this because I don't understand a lot. I kind of understand the general things about it, and then I'll go into my work on the waiting time problem. Now, this waiting time problem was mentioned in passing by Dr. Dan, and that was a great. I only got to see part of the show he had with Dr. Michael Bechly, and it raised a lot of interesting questions and perspectives. And in the side chat there, I was saying, what about zinc fingers? What about zinc finger proteins and the waiting time problem? So we'll cover a little bit of that. And so I'm going to have to wing a few things here. So let me look for the journal Theoretical Biology, Ola Hossjer, journal. I'm having a little bit, Google's giving me a, let's see, waiting time. Okay, I'm going to read it. I'll post it here. Just take it for what it's worth, because if you don't completely understand it, neither do I. Dr. Dan has read this, and to his credit, he has at least read the whole article. I know enough that it'd probably take me a week to understand, really appreciate this, because I haven't worked on the waiting time problem. I've been I don't often, I'm pulled apart in so many directions these days. We're all spread really thin in the creationist movement, and I have a lot of irons in the fire. This would be an interesting problem, but I've decided to focus more on kind of the area of abiogenesis and eukaryotic evolution. The waiting time problem has been used to talk about human evolution and other kinds of evolution, and that's just something I was reluctant to get into because it's really difficult. What we would call the benefit to cost is not really great, at least as far as my skill set. Whereas dealing with abiogenesis and critiquing it and dealing with eukaryotic evolution, the benefit to cost ratio was really good. You could get a lot of mileage without having to expend as much effort. So let me go ahead and try to bring this abstract up. Oh, I might even be able to get a PDF. That'd be even be better. Let's see if it will give me a PDF. Hopefully my system won't crash. Oh, and Godzilla Freak says, your boss handles the waiting time problem very well. Okay, just speaking of Dr. Sanford and my role in this, we're going to talk about zinc fingers. And when I was reporting on my job when I was working kind of as a on-site reporter for or discoveries at the NIH by attending classes there and seminars, and I report to Dr. Sanford back what I discovered, because we were looking at just incredible features of the genome. And the zinc fingers came up just kind of in my routine searches. I said, hey, John, look at this. And as I was explaining at it, he said, we need to do a paper that explores waiting time with these zinc finger proteins. And that's where we left it. I'm working with Dr. Sanford now, trying to get a paper that will mention zinc finger proteins, and we'll cover a little bit of that today. I'm a big proponent of open review, and it's been very helpful. So I don't mind sharing what I'm researching a little bit, because I'm not an expert in these fields, and it's been helpful when someone who might be a hostile entity and say, Sal, your ideas are just trash, rewrite this. And then I do it and they say, well, I really can't object. Your work still sucks, but I have nothing to say anymore. I mean, I've gotten that treatment, and I'm usually very grateful. The feedback has been accurate and the paper gets better. And even one of my best works, you can actually go to Dr. Sanford's website. You'll see it. He actually lists it on his website. It's co-authored by me and him on nylon-eating bacteria. And editors have told us our conclusions were correct, but they're just not going to publish it. They give a variety of what I think are silly reasons, because we are attacking the work of a National Academy member who's very revered, in Susumu Ono, the guy who coined the word junk DNA. But thankfully, one editor of Cell Reports, and Cell is a prestigious journal, very respected. Their open access Cell Reports, he was the editor-in-chief of that. He affirmed that our conclusions were right. He affirmed that publicly on Swami Das's website, of all things. Why did that happen? We think Dr. Swami Das is trying to curry a little favor, get an endorsement from Dr. Sanford for his genealogical atom book. And he was so eager, Dr. Swami Das pulled some strings and got editors to try to have our paper pushed. And well, that never happened. So that's just a little backstory how that ended up. That's just a little backstory of how that all played out, that we actually got an editor of a major journal to affirm our work publicly, even though he's not, he said he's not going to help us publish it. But that was a big endorsement for our work. So I don't know how I got on that topic. Oh yes, I was talking about open review. So I'm going to throw out some of the stuff on zinc fingers. Now, the publication I'm working on is not a waiting time problem. We may mention it in passing, meaning we'll say, this is the architecture of zinc finger, and it may raise waiting time problems. That'll be it. That'll be it. And we'll put the associated references. So that's how. So thank you very much. Who said that? That was someone said your boss did a good job. Yes, Godzilla Freak. So thank you very much for the kind words from my boss. I'm totally indebted on many levels, and my family as well, to Dr. Sanford. While my mother was needing 7 years of 24/7 care, I was able to work from home because of him. So not just my mother or myself, but the entire family is very indebted to Dr. Sanford. Not to mention that was a great job. Fabulous. So anyway, let me get this going here. So from the Journal of Theoretical Biology, on the waiting time until coordinated mutations get fixed in regulatory sequences. Department of Mathematics, Stockholm University, Stockholm, Sweden, the Biologic Institute. And so I would guess that would be Gunter Bechly and Gauger going through the Biologic Institute. I'm just going to read the abstract because this will tell you, just like what the rest of the paper will be. Sometime, I don't know if I'll ever get around to it, actually get into the math of a waiting time problem. You know, and the more persnickety details. I'm sorry for that. That's really obnoxious, this thing demanding feedback here. I don't know why it's... How would you rate? That's going to be terrible. And I don't know who's making me try to submit feedback. Who's asking me for feedback? I don't know what's causing that. So I'm going to kill the PDF there. All right. Oh, there's a debate going on. Oh, well, this is a nerd session, so I don't know if this is going to be your cup of tea. We're going to talk about zinc finger proteins and transcription factors. So anyway, let me... I can't seem to make it bigger. Anyway, in this paper, we consider the time evolution of a population of size n with overlapping generations in the vicinity of M genes. We assume that this population is subject to point mutations, genetic drift, and selection. More specifically, boy, I'm having a huge delay here. Apologies. More specifically, we analyzed the statistical distribution of the waiting time, Tm, until the expression of these genes have changed for all individuals. When transcription factors recognize and attach to short DNA sequences, binding sites, within regulatory sequences in the neighborhoods of the M genes, the evolutionary dynamics is described by a multi-type Moran process where each individual is assigned M times L regulatory array that consists of regulatory sequences with L nucleotides for all M genes. We study how the waiting time distribution depends on the number of genes, the mutation rate, the length of the binding sites, the length of the regulatory sequences, and the way which the targeted binding sites are coordinated for different genes in terms of selection coefficients. These selection coefficients depend on how many binding sites have appeared so far, and possibly on their order of appearance. We also allow for back mutations whereby some acquired binding sites may be lost over time. It is further assumed that the mutation rate is small enough to warrant a fixed state population so that all individuals have the same regulatory array at any point, at any given time point, until the next successful mutation arrives in some individual and spreads to the rest of the population. We further incorporate stochastic tunneling, whereby successful mutations get mutated before their fixation. A crucial part of our approach is to divide the huge state space of regulatory arrays into a small number of components, assuming that the array components varies as a Markov process over time. This implies that Tm is the time until this Markov process hits an absorbing state with a phase-type distribution. A number of interesting results can be derived from our general setup. For instance, that the expected waiting time increases exponentially with M for a selectively neutral model when back mutations are possible. And I'm like, oh my goodness, that was thick. I need to go take some Advil, if you know what I mean. So, guys, do you want me to read that abstract again and try to uncoil a few parts that I understand, just speak up, or you not forever hold your peace. You can speak up, and otherwise I'm just going to move on straight to the zinc fingers. So this still is a victory for the ID movement and creationists. These are the sort of peer-reviewed papers that are quality. We may not say Darwinism is wrong, but we will still take questions that the evolutionists themselves have put forward. Yes, the abstract was as clear as mud. Still take ideas that evolutionary biologists themselves have put forward and say are problems, and we just elaborate it. Oh, and here it is. Good question. Okay, what relevance does this have to intelligent design? Simply put, and just collecting, there have been many papers written about the waiting time problem. I wouldn't say many. I know of about 10 offhand that I've seen, maybe 5 offhand. Durrett and Schmidt, Sanford et al., this one, and it's been mentioned in a few others. So I know less than 5 offhand. They're probably, I would just supposing, maybe 10 publications, because this is a very specialized topic. And what relevance does this have to ID? So if you're familiar with the idea of irreducible complexity, and you know, people who know me, I don't like using that word. So let's just say, if you're familiar with the idea that you need many simultaneous changes to make something work, there's a difficulty in evolving that. So this is much like a password. If you have a short password, if you have a very short password, say 1 letter, a 1-letter password, a hacker could easily break that. You just, you know, let's say it's just lowercase letters of the alphabet. Well, all you have to do is just make 26 tries, you're going to break the password. However, if you have a 2-character password, you'll need 26 raised to the 2nd power tries to break the password. If you have a password that is 10 characters long, and assuming again lowercase alphabetic letters, it's 26 to the 10th power. Once you get up to a 20-word password, it starts to be like 26 to the 20. These start to be very large numbers. So I'm just giving you a feel how quickly it is for a feature to work that you need these simultaneous mutations. What Michael Bechly did, and I like the idea, I just don't use the word irreducible complexity, because then you get in problems of defining it. If I just say you need these simultaneous changes to happen, you need all these parts to be there at the same time for this to work. I gave some examples where natural selection is going to have a very hard time, and no one has been able to refute it. I've said, okay, tell me how topoisomerase evolved in incremental steps, and I'll identify all the problems. If some of you have heard this, if I replay it again, we're going to just eat up more time trying to talk about it. But no one has refuted. I kept putting it on the table, and we know it's, I'm pretty confident no one's going to refute it. You need all the parts. A half-formed type topoisomerase is deadly. Topoisomerase cuts the DNA, and it also puts it back together. If you have a half-formed topoisomerase that only cuts the DNA and doesn't put it back together, it shreds the genome, the organism's dead. And we know the effect of disrupting topoisomerase is deadly, because that's what we do in chemotherapy. We give them chemicals that will disrupt topoisomerase function, and it kills cancer cells as well as good cells. You just try to, it's kind of the nuclear option, and you just hope you don't kill the individual in the process of throwing all this chemotherapy that's attacking topoisomerase. So I'm pretty confident the idea of simultaneous changes and all the parts needing to be there and in the right place, not just being there, but in the right place. James Tour showed a picture of having all the parts, like say of a Volkswagen. It's not going to do any good if you don't have any way of putting it together in the right order and in the right place. So all these claims by evolutionary biologists that they've solved the problem because they do a phylogenetic reconstruction, it's like, nope, you need way more than that. And so going back to Red Barrel Rider's question, what does this have to do with ID? So it first starts with the question, how hard is it just probabilistically speaking? So you just say that, how many parts do you need? So in here, let me see, they allude to it a little bit. When they say M genes, change the expression of M genes. You'll see it right here. It says change the expression of M genes. So M genes. So this is, so if we have to change M genes, but even really within the gene, we might have to change individual parts, and I'll cover that with the zinc finger. So just imagine the critical parts of the protein need to change like a password. So that's why when, and it alluded to this, let's see, and this really should be obvious, let's see, exponentially, you'll see it here. I'll try to highlight it. It says, a number of interesting results can be derived from our general setup. For instance, the waiting time increases exponentially with M. The waiting time increases exponentially with M. So let's just go back to the password analogy. Say you've forgotten your password, and let's say it's a small one. You can kind of fiddle with it. You know, it's like, did I need to add this character or capitalize this? If it's not too complex a password, you might be able to solve it. But if it's a really long one, and because of computer security issues, people are writing longer and longer passwords, and it's becoming easier and easier to make mistakes in remembering your password and then typing it in. You could see, or let's just put it in another way. If you have a very short password, an intelligent hacker will have an easier time than if you had, like, say, a password that's 20 characters long. Now, I'm being excessive here deliberately. He'll have an easy time with a one-character password. So there's no reason to do that. I've had, like, you know, some of you have like 4-digit pins. That's more like a 3-character password. Okay, it's on the order of that. So the idea of waiting time is how many attempts do you get? How many resources do you have to expend to solve the password? And I'm going to address, you know, Dr. Dan astutely said, although I believe he's wrong, and I have, you know, so I have to disagree with my distinguished colleague, Dr. Dan Stern Cardinal. He was astute to make the objection that evolution has no direction, therefore you don't use the word target. That is correct. The correct way to frame it, and they did use the word target in this abstract, but they got the right idea in the title, coordinated. And maybe if I could show a visual picture of this, the difficulty of coordinating. I'm going to try to bring up, see if I have my debate with James Downard. Actually had the picture that I want to show, and I hope I don't blow up my computer in the process. So there are various ways to frame the idea of coordination. And if you'll forgive me, I'm not going to do a slide, put PowerPoint in full slide presentation mode, because it makes it a little difficult for my computer. This is just an editing mode. So we could say that when I lay out these coins, it's in an uncoordinated state. Like, say if I had a tray and I just threw these quarters on there, could put it in an uncoordinated state. This would be viewed as a coordinated state here. Or even better here. You'll notice the quarters are all heads. They're all arranged in nice rows and columns. And the George Washington's bust is all pointed up in the up direction. So you can use the idea of coordination. Hopefully you understand that this is a coordinated state because each part is coordinated towards some recognizable pattern that's not, that can't be said that we made the pattern after the fact. The pattern I showed you is a violation of the law of large numbers. And it was really nice to hear Dr. Bechly in his discussion with Dr. Dan on July 7th on Dr. Dan's channel. Dr. Bechly used the word law of large numbers. So seeing all those coins heads is a violation of the law of large numbers. When you have violations of the law of large numbers, that's one way we could, qualitatively speaking, I'll call that coordinated. Another way we could say something is coordinated. So let me show a picture of this. This is uncoordinated. See, these cards were randomly picked out of a deck and then just thrown randomly on the porch, for all we know. Yes, it's an astronomically improbable configuration in that you're probably not going to be able to repeat this, even to get it exactly that way with those cards. And, some are flipped up, some are flipped down, and the kind of the orientation relative to each other. But one wouldn't be astonished, one wouldn't infer design. However, we would definitely infer design here, and more so here. We don't have to say target. See, we can avoid using the word target. But I mean, in a sense, like with the card here, one card has to be kind of positioned as a target, and then the other one kind of has to lean up against that target. So these are just, again, we're splitting hairs in terms of the semantics here. But if we stop using the word target, then I think that'll deal with Dr. Dan's objection, which I feel is more of an objection based on choice of words rather than substance. So I'm just pointing out we can use different language. We could use the word coordinated, and they thankfully used the word coordinated in the title. So we don't have to say a particular site is a target. We could say it's coordinated with another molecule. And I'll show another form of coordination. Let me see here if I have it. Do I have it? Yes. So we could say that a key is targeted to a particular lock, or vice versa. Usually I'll think a key is targeted to a particular lock, but we can just avoid that word and say, what's the likelihood that we could have a key that is coordinated to open a lock? The end result is that it will fit and it can open the lock. We don't have to say target. What's the chance a random process will make a complex lock and key combination from just an assemble of random metal molecules? And you can see that this is, even if you had like tornadoes passing through a junkyard for a long time, it's not going to make a new lock and key system anytime soon. How many times do you think a tornado would have to pass through a junkyard before you get a new lock and key? If you want a new lock and key, you hire a locksmith. So that's basically a long answer to a very good question from Red Barrel Writers. And let me look at the side chat to see if that Godzilla Freak said that's a really good analogy. And what do you mean by work? In the context of evolution, we're talking about reproductive fitness. No, we're not. Yeah, for some reason, zinc finger sounds like a deadly disease. I like the idea of having a password where you just repeat a character enough times that it's outside the range of what anyone else would guess. Okay. So again, what password abstraction has to do with zinc fingers? That's at the end of the talk because I have to introduce the zinc finger architecture. It's going to take about an hour to introduce the zinc finger architecture. By the way, nice to see you, Creationists. And I know he's on the other side of the world, so this is great. So good questions, and I'm glad we have at least an audience. Couldn't two fingers grab a molecule or atom? Yes, you'll actually see that with a zinc finger. The zinc finger protein will actually, its zinc finger domain will actually grab the zinc ion. Oh, let me see if I'm having, yeah, Restream's a little bit, it does have delays. Because I've had very good discussions, Creo debunk, I want to look especially what he has to say. Okay, so we must discuss in all detail that there is no functional gradation there really. There may be simultaneous change of different components, what later creates the image of and simultaneous creation. Imagine a pair learning how to dance. Okay, and immutable destinies here. Okay, so guys, I really do want to respect her. I can't tell you how grateful I am that immutable destiny and Creo debunk here. I have greatest respect, and I don't mean to minimize your questions, but I had a pre-programmed, kind of a pre-programmed show that I slapped together, and I'm just going to present this also for the benefit of those who are maybe not as familiar with molecular biology as you guys. So, but the idea, and you'll see that I'll go back to this lock and key analogy. Granted, in biochemistry, we don't like to use that word anymore for protein interactions. We like hand in glove or induced fit and terms like that. But just suffice to say, let it be just kind of a metaphor for the uninitiated. It's good enough for our purposes, or like password that corresponds to a login. So like I just realized when I'm loading up a PowerPoint, it will slow down my system tremendously. So let's just, hopefully we can just slug through the architecture of zinc fingers, because that paper specifically mentioned transcription factors. A lot of transcription factors have zinc fingers in them. And so this is totally relevant to the paper that that the creationists just scored in. So there was a presentation I made to faculty, and we had, through various contacts, there are groups of us that are people that are professors at either secular or Christian colleges that are ID or creationist friendly. And sometimes in meetings I've had the privilege of presenting a few times before a nice group of them. They would include deans and faculties, members. And the presentation I'm about to give is one I gave to the deans and faculty members. Some of this they would already know, but maybe out of their specialty. I got good feedback on it, and when I criticized evolutionary theory for some of the ideas that they had, they all laughed. I said, okay, they get it. All right, so this slide has been kind of redone. The slideshow has been redone a little bit, so if it's messed up, that's definitely on me. I had to adapt it from the presentation I gave like a couple years ago. And some of this may be already old hat to some of you, but I'll just go through it here. Let me see if I could minimize this, and I could make this a little bigger. So the original title of my talk was Exploring Promiscuous Domains, and I'll explain what that is. Exploring promiscuous protein domains and DNA protein motives through pedagogy and practice. And the example in all of this was the zinc finger protein. But I open up with collagen because that's an easy one to understand. So if you go to Walgreens, you can find like beauty products like this one. It's a collagen beauty builder. Collagen is a real protein. It accounts for about roughly 25% of the protein mass in our body, which is a lot. So you drain out all the water and fats and other chemicals, and you just have proteins. A quarter of that is collagen. We have the amino acids here. There are 20 canonical amino acids, and they have various properties. You could see it described here in the Venn diagram. So you could see like one of the big circles here is like the hydrophobic, and then so that's very important. And you have some like what we call the aromatic groups, and then there's this hydroxylic, and then you have the aliphatic. You'll see why I'm pointing this out. So we know proteins are made of amino acids. You just take the amino acids and make a chain with them. And you could spell collagen. This is collagen type A1, collagen 1, collagen, A1, collagen A1 in humans. It's collagen 1, I'm sorry, collagen 1. There are like 28 some collagens in humans. And in addition to all those 28 major families, many of those families have paralogs within the family. So collagen 1 is a family made of two members, collagen A1 and A2, or sometimes collagen 1 alpha and beta, where alpha is one paralog, beta is the other. So that's neither here nor there, but you could see the identifier here. I got this from Uniprot, and that's a spelling of the collagen protein. Now, I'm going to highlight a very interesting feature here. Isn't that amazing? That's a non-random pattern there, highly organized. And it's an interesting debate how difficult this might be to evolve a collagen. Now, it may look trivial enough, but let me see if I have a picture of this. If I actually covered the collagen in all the parts, it is just mind-boggling. Mind-boggling. All the post-translational modifications involved in collagen, not to mention that the collagen protein will actually split into 3 separate proteins that have separate functions. So it starts out as one functioning whole and then ends up being 3 functioning parts with independent missions within the organism. So don't be, at first the evolutionary biologists may say that's kind of trivial, you just need some sort of tandem repeat. It's way more complex than that. Way more complex. But that's subject for another discussion. So I call this skittleizing. Dr. Sanford and Josiah Seaman created what they call DNA skittle. I adopted it, that convention here in protein. So I call this skittleizing when I throw in colors. So you can see the pattern there. And so we have what we might call an important motif in collagen, where every 3rd amino acid, or we use the term residue, is a glycine. So we have this motif. It's easily seen here. I'm just showing it. And so we have we have protein motifs, and then I'll talk about protein domains, and then we have DNA motifs, and there are a variety of those. We'll go there later. So I'm just saying, when I talk about motif, they may be in a protein or a DNA. In the case of collagen, the obvious motif is this one where you get a glycine every third amino acid. Any questions? Collagen A1 is heterotrimeric, actually. So that collagen A1 is the collagen 1 is a heterotrimer. So it is 2 of the alpha and 1 of the beta. So I'm just going to look at the side chat if anyone had questions about this. And I'm going to criticize also this thing about the supposed stepwise evolution of these is going to be problematic. And I'll show the mechanics of why. That's why it's going to be a long lecture. So thank you for the comment. Nonetheless, I'll show why it's erroneous. And one of the many reasons I think evolutionary biology is just generally bogus. They never think through things carefully enough. Kind of like Dave Farina on steroids. But in any case, let me talk about domains. So first I'll define it, and if it doesn't make sense, maybe you'll kind of visually see it. A protein domain is any identifiable subsequence of a protein that can fold, function, and exist independently of the rest of the protein chain or structure. So we may have the whole protein, and then we have parts of the protein. So I'll just move forward, and maybe you can see it. Maybe it'll be easier to just visualize it. And so what motivated some of this was this paper, Evolution of Protein Domain Promiscuity in Eukaryotes. Now this is really kind of, I have a funny backstory here today. Okay, we call these promiscuous domains. We call these promiscuous domains, promiscuous protein domains. I wanted to actually get a thumbnail to highlight, to kind of get the image of promiscuity. And I Googled it. I typed in promiscuous, and then I looked for images. I got some funny hits. Okay, it's not very family friendly. I said, I better kill that idea. Okay, so I'll talk about what promiscuous domains are. I was like, I hope I shut the browsers that gave me those those images. But this paper, Evolution of Protein Domain Promiscuity in Eukaryotes, that actually relates to the evolutionary problem I'm going to highlight. Some of the evolution of zinc fingers would require promiscuous movement. Sorry about that. I don't know why they want to just use derogatory words to describe God's creation. So, they'll talk about junk DNA and then promiscuous domains. I mean, couldn't they give it some title that's more design friendly and would talk about the glory and majesty of the creator? It would suggest it. And so anyway, I'll move on. So you'll see the names here that include Eugene Koonin and Igor Rogozin. And Rogozin was my professor of bioinformatics. So that was my that was my brief introduction to evolutionary phylogenetics. It was in the context of a bioinformatics class, of which phylogenetics was a very small portion, but he's a computational biologist who worked with Eugene Koonin. So now we go to a human crab zinc finger. It's 540 amino acids, and Well, thank you for this, and I do want to. It's great because it's great that I do face critics that actually are thinking, and I could see by your comments these are thoughtful discussions. So you might have to wait till the end of this to see how it supports intelligent design. And I will mention that that is usually a regular talking point. It's like, do you have a mechanism? Can you describe the manufacturing process? Is that required to believe that a cell phone is designed, that you have to be able to detail the manufacturing process? Yes, it's easier to believe a cell phone is designed because we know humans can make them, but we actually don't need to have details of the chip manufacturing process or how the plastics were fabricated to know that a device is designed. So what we use in mathematics is that it's sufficient to have the mechanism, but it's not necessary. Sufficient, but not necessary. So does IB supply a mechanism? I'll say no, not in the traditional sense, because intelligence itself is not really well understood. It may actually be fundamental and primitive to the universe, and therefore cannot be described in terms of physics and chemistry. It might actually supersede that, but that's neither here nor there. But anyway, this is the zinc finger protein. Now, if the fonts are a little hard to read, just accept that there are letters there. Let's see if I can make this larger just to help the readers. Hopefully that helps a little bit. Do any of you see a pattern? I'll show the pattern here. Okay, so you see the pattern here? The dark red is the cysteines, and the purples are the histidines. They have to be roughly separated by about these proportions. There'll be some variability, but this is very important for the biophysical properties. Someone asked, can it actually grab a single atom? It can grab a single ion. If it's spaced correctly, it's very important to the fold. And so in the thumbnail, we had that picture of a zinc finger with, it talked about the alpha helices has the cysteines, the histidines are from a beta sheet. There's a beta sheet somewhere here. When you arrange these molecules together, it forms a beta sheet, what's called a beta sheet. And then somewhere around here, it'll form an alpha helix. I'll actually show a diagram of that. So, all right, so I'm going to show a little more there. And now I'm going to add coloring that follows from the Venn diagram there. So now, not only specific residues, but maybe the residues in terms of the families. You really see kind of the organization there. So hopefully that makes it a little clearer. And you can see this is kind of the general motif here. Now there are various, depending on what literature you read, it'll describe what makes the zinc finger motif distinctive. Just to time out a little bit here, some of you may have heard discussion. I said, how can you have common descent from one single ancestral gene slash protein, gene slash protein? You can't, or you can't prove it, because you could see that the architecture of zinc finger is very different from a collagen. You can even just see it just in terms of the letters. There's not a slow stepwise evolution to go, where you could have a common ancestor that evolves into a zinc finger, or to a collagen, or a topoisomerase, or a helicase. And Dr. Dan and Immutable, others have said, yeah, we don't expect common ancestry from a single gene, even though they generally believe in some sort of common ancestry from an organism. But even some evolutionary literature is now kind of euphemizing the term from a single cell. They'll say, oh, LUCA was a population of organisms, which means maybe, so there are words like FUCA versus LUCA, first universal common ancestor, and LUCA is the last universal common ancestor. Was FUCA just one creature, or was it a population? Because if FUCA is a population of organisms, then you don't have universal common ancestry, period. But let's assume for the sake of argument we have cellular universal common ancestry, that there was one organism, even though, like I said, notwithstanding that this is starting to become in doubt, we don't have a single ancestor for all genes and proteins. So even if we hypothetically evolved one protein that was functional, it can't be the ancestor of all the proteins that are extant. They would have to be piecemealed together and kind of poofed synapomorphies to make all these parts. It'd be a strange thing, and I'll cover a little bit of that. Oh yes, and here was my point. Let me highlight it here. You can actually see it visually. You can see the collagen on the left and the zinc finger architecture on the right. It's unreasonable to expect that this will you can't throw this in a phylogenetic software. Even with all the extant proteins, you're not going to find a common ancestor. There have been various attempts to do that with some of the software as they're trying to classify all proteins, and it usually fails to unify it under one architecture. And for that reason, most biologists I ask will say they don't believe it has a single ancestor. It will have to have multiple ancestors that share parts. And that would have to be in my opinion, that would have to be kind of miraculous, that it'd have to be kind of miraculous that it would share parts and you'd have a functional protein. You have to have pieces of proteins, or dare I say domains, kind of jump all around and form something new. And that's why it's the word is promiscuous domains. And even in that paper that I cited, they said this is a mystery we haven't solved. Of course, the creationists will say that's because God did it, and there's no mystery, because evolutionary processes are incapable in terms of physics, chemistry, and statistics to make such functional systems that have quote-unquote promiscuous domains. So what I would say is, so you can kind of see here you would need macroevolutionary changes, and then just by way of metaphor, we could say that we have architectures for pianos, automobiles, and helicopters. They can be variable with kind of macro evolutionary or macro or micro differences within the kind, but then you can't really evolve one kind to the other without breaking it. And hopefully I made my point. You can't evolve a collagen into a zinc finger without breaking it, or vice versa. And then similarly, it's hard to imagine them descending from a functional common ancestor. All right, so now let's look at a little bit more about the zinc finger. See if I can. So for lack of better metaphors, I'm going to use the word key. Maybe it's better to say it's a lock. And so if you all have better metaphors you can suggest, I'm open to it. So there are various ways to represent the fold of the zinc finger domain. So each line is a valid C2H2, which is cysteine, 2 cysteines, and 2 histidine. They call it C2H2 zinc finger. That's just kind of the terminology that's used. We call that the classical zinc finger. There are various kinds of other zinc fingers. So if we spell each line would make you can represent it in a two-dimensional fold like this. where you have the zinc ion connecting to the two cysteines and two histidines, or you can have a 3D diagram like this here. And this is a Richardson ribbon diagram. So when you see this ribbon, there are actually amino acids here. Each line is a valid C2H2 zinc finger. And maybe I'll cover this a little more. So let me just show a zinc finger here. And I'm going to try to animate, halfway animate. I'm going to turn it on its side. So I'm turning it on its side deliberately, and you can now see each of the lines here is a separate zinc finger. Each of the lines here is a separate zinc finger that looks like that. So you have actually an array of zinc fingers. The other thing is, and I'm going to just highlight the point again, where the cysteines and histidines are. So each line has its own, it'll have to grab its own separate zinc ion. Okay, and let me look if there are any technical questions specific to what I'm discussing. Okay. All right. I don't see anything specifically to the zinc finger question. I'm glad for all the questions here. There was one question about amyloids or something. You can tag me. I may have to just keep moving on. But if there are questions specific to what I'm presenting here, I'll try to answer those, because in case I'm not communicating this well, I need to know that. So apologies in advance if I skip your questions, if it's not immediately related to the presentation at hand about the mechanics of the structure and function of zinc finger proteins. So again, I said each line is a valid zinc finger, and this was kind of, the next one is a thumbnail from, this is from the thumbnail. That's why they call it a zinc. Okay, maybe it's a good time to pause why it's called a zinc finger. First off, let me just minimize this a little bit. The reason it's called a zinc finger, first off, zinc, it has the zinc ion that helps the fold. One reason it's great to have the zinc is the zinc ion there, just from principles of structural biology and protein folding. That little piece of glue, so to speak, enables us to make a nice, enables the protein to have a nice shortcut, because otherwise you'd have a very complicated protein with lots of amino acids, but you can make it even smaller if you kind of cheat, so to speak, and allow things that are not amino acids to help build the protein. So that's where you get the word zinc from finger. And the reason, at least why, whether this is apocryphal or not, but it's the adopted story, is that the reason it's called a finger, it's like a finger that can grab, and it often will grab DNA. So this protein actually, if you imagine the zinc finger protein, just imagine it like a hand, and it's going to grab onto DNA. But it's more complex than that. It's going to identify a specific section of DNA and then connect to it. So that's at least the story of where it gets the name zinc finger. And so I really like that picture here of a finger to describe the function of a zinc finger. That picture was, whoever the artist who drew it was very ingenious, because it captures the spirit of what a zinc finger protein does. So we'll just continue here. There are other kinds of zinc fingers that are non-classical. We used to believe they're only in eukaryotes. There is a very different architecture of zinc fingers in prokaryotes, radically different. It's only recently we have realized them. And this is a good observation here by Immutable Destiny. The interaction between the metal and the atoms coordinating with it is stable enough of a complex to allow the rest of the protein to do other things. So correct me if I'm wrong, Immutable, that this is an example of a metal binding protein. There are other metal binding proteins like think hemoglobin. So they're proteins that will have parts of them that in order to function, they'll have metal in them. So there are others, just for the record, there are other kinds of zinc fingers, but we're focusing on the classical zinc finger here because it's the easiest it's the most well studied. It's the most well studied, and it's the easiest to understand. It's been interesting for people in biotech to pursue it. Now, I'll point out something. So if I will say just a little bit of terminology, I'll say each line is a valid domain. So remember the idea of a domain is kind of like a discrete part. You can kind of subdivide the protein into parts and say that's a domain that has an individual function and fold. It can almost be described in isolation. So that's what they mean by domain. And there are other domains here, and I'll identify them by the color coding there. That is the crab domain. Now it's not as recognizable as the zinc finger. The zinc finger domain you can almost visually see. The crab domain requires computational and maybe even biochemical methods to identify it. It's like a separate component. I bet there is a goldfinger. I think there may, that's an immutable destiny. To find out if there is, I don't know, try goldfinger proteins. Just Google it. That'd be interesting. They're, they have things like the ring finger. They have all sorts of names. So they're at least, so you could see there's several domains here. There are like 13 zinc finger domains, one crab A domain here. The crab A domain has a fold that looks like that. So I showed you the zinc finger fold. This is the crab fold. And so I'm going to say differences in zinc finger sequences are functionally important, much like notches, slots, and grooves in a key, even though the keys share a common architecture. So you could look at a key like the keys here, and you could say, yeah, they have a very similar architecture, but they're not exactly identical. Likewise, if you look at each of the zinc finger domains here, you could see the spelling has these two C's here and the two H's. It's very distinctive that makes the zinc finger domain. But other than that, there's some subtle differences there. And this is like changing notches in a key. So it has the same architecture, but there are differences in the architecture. Now, evolutionary biologists will say, well, they're just microevolutionary changes from some sort of domain duplication. And I'm going to point out why this is a problematic hypothesis from the biophysics of it. So what my evolutionary colleagues will say is, well, you just start out with one zinc finger, and then instead of a gene duplication, you have a domain duplication. And there are mechanisms that will allow these sort of tandem repeats or like homologous recombination. Any sorts of mechanisms are proposed. But let's just say, grant for the sake of argument, temporarily, you could just duplicate this and then kind of duplicate this pattern and then just kind of modify it a little bit until it's functional. And that actually, as you think about it, it's going to pose problems for evolution, even though superficially it would look like all of these domains are homologous, and you could build phylogenetic trees with this, and I've done that, and I'll show that. It still will not solve the fundamental problems. Speaking of which, so if I took all of these domains here, these individual domains, and put it in a phylogenetic program, you'll build this tree and say, oh, that's proof it evolved by common ancestry. And now I'll pause because CreoDebunk threw in a really good objection here. Are really duplications necessary, or was it about just preferring functionality of some part of the gene? So just gradual evo without duplication. The answer is duplication won't do it. So we might say it's necessary, but not sufficient. You can't evolve them one at a time. You have to have several zinc fingers to get the necessary specificity. And so as I go through the lecture, you'll see I'm going to defend that thesis. What I just stated, you'll say, at this stage, I haven't defended that thesis. But I'm going to try to unfold this, and you can look at it and ponder it. And when Dr. Sanford saw this, he said, this is a good thing for waiting time. We can look at it. And then, unfortunately, I got pulled off to other projects. Still was involved in the zinc finger, did a lot more work on the topoisomerase and stuff on population genetics and other things like the nylon-eating bacteria and even more things beyond that. Didn't have a chance to incorporate the zinc finger problem into the waiting time problem. But it's still preliminary in that you could actually see some of the principles, because zinc fingers serve as transcription factors, which were the centerpiece of many of the waiting time papers. So this is actually getting down into the biochemistry and the structural biology of this. It's a very good question there, Creo. So now we'll just move forward. So I can actually point out here, if I took this phylogenetic tree and I'm going to relate it to the individual zinc fingers there. So I'm just going to pause here and let you look at this tree a little bit. Not to mention, it took me a really long time to build it, where I had to draw the colored arrows to point to the zinc fingers. So you could see how this tree diagram would have to play out, how the duplications and then subsequent microevolution to each of the parts. Now, this relates a little bit to the discussion that I've had on discussions, plural, I've had on this channel about phylogenetic methods. We can kind of guess maybe which pieces are closer to each other and say this evolved from that, this evolved from that. But then depending on how you root the tree, you might get a different story. So, but let's just say hypothetically that I believe this is a mid-rooted phylogenetic tree. Let's say hypothetically this is close enough to if you're reconstructing an evolutionary history. So we could see then that we have this one very similar to this one. So this one here and this one here split off. And then let's see another one that's extreme. So then this one here and then this one here split off from the same common ancestor. I don't know how you're going to build this, okay? But let's just say hypothetically we start off with a tree that tries to evolve this zinc finger from these 13 zinc fingers from a single zinc finger. In their various permutations, we could do this. We could say, okay, let's just assume this came from a totally different already formed zinc finger and we're just tweaking it. So we'll have to take various evolutionary routes to how this evolved. And once we start thinking of this and the biochemical implications, the challenges start to appear, even before we get to the waiting time. So I'm going to pause just a little bit here because this is I have to disagree here. Functionality should be thought of as a gradient, not as a binary thing. Unfortunately, it's very close to binary. It may have some gradients, but there's a, let me say, what's the proper word? It's not a nice spectrum. It tends to be one or the other. And if it's not, that's not a good thing. So it could be. I'm not saying it never is. It could be, but there are many situations where this is bad, and I'm going to highlight those. And Immutable actually did point to humans who are trying to engineer these. When you misfire in doing this and you try to say it's half functional, what happens if the zinc finger half functions and it grabs the wrong piece of DNA? That's very problematic, and we'll cover that. So I appreciate the objection, but let's just keep going through this a little more. Oh, and Ray here, also our resident chemist. How is evolution doing all this selection and preservation without any intelligence, goal, or knowledge? It is, by the way, the zinc fingers that are human-made, they're having a bear of a time making them as good as the God-made ones. Just a point of there. We try. We can't make them anywhere near as sophisticated as some of the ones that we see that are God-made, like the transcription, the TF, the transcription 3A. It's very, very sophisticated. We can't even come close to it. So when we hear about James Tour talking about the chemistry, that the chemists are having a hard time synthesizing these chemicals, we're having a hard time even just designing. You know, we can make artificial proteins, but we have to actually even think. We have to be able to think. It's like, okay, we don't have so much of the chemical problems because we can, have ways we can genetically engineer all sorts of things now. But the problem now is one of design. Can you make a zinc finger that's going to target a very large section of DNA? We can maybe target 9 bases. To have something like transcription factor 3A, they can simultaneously, in one state, bind to the 5S ribosomal DNA / RNA, maybe to something like 50 bases, and then also 50 bases of DNA, and then a different mechanism to bind to many bases of RNA. Just incredible. We couldn't do that. Beyond our knowledge of biophysics to build something that sophisticated. We have, we get successful to about 9 bases, but even then not too good. We don't have, you have to do a lot of trial and error. So I just want to acknowledge that great comment. And then also, Sal, the kinematics associated with enzymatic don't always permit for the protein to be functional at all times. In fact, you don't want it to be, you actually want the protein to shut off every now and then, and that's actually another important point. So we'll continue here. Thanks for the great questions. Godzilla Freak, if I'm being too repetitive, I'm sorry. All right, so I'm just going to move on here. So people are asking, they're like, Sal, how can you be taught phylogenetics and you disbelieve evolution? I'm showing you one of the reasons why, right here in the zinc finger. And we've been going an hour and a half, okay. Making good time. This could be a tough lecture. Okay, so again, going back to the zinc finger, we have the crab domain. There's ways we can represent the domains here. You could see here, I'll just use the word crab, and then I'll just use multiple zinc fingers. It's kind of hard to have 13 of the little boxes here, so it's going to be fewer than what's actually there. But this is just kind of a qualitative representation of the zinc finger. So let me just show that representation. And I'm going to highlight this alternative representation here. So you can represent the protein in terms of its domains. You have the crab A domain plus some other residues, and then the zinc finger domains. Now, there's pictured, let me see here, count 1, 2, 3, 4, 5, 6, 7, 8. There should be 13, but I just grabbed this picture. Is a close enough rendering. So we can have other kinds of zinc fingers. When this crab zinc finger, it'll be actually cleaved, that is, cut in pieces. And so the crab piece here will be cut out, and they'll kind of have a life on its own. So the zinc fingers by themselves will do something, and the crab will float off and do something. And there's a reference to that. It's really amazing. Oh, Godzilla Freak. Oh, no, I was referring to when you said mid-rooting. If you had a question on that, feel free to tag me, and I'll try to look if you have a specific question. It's not that critical, whether it's mid-rooted or whatever rooted, it's not that critical to where I'm trying to head. I'm just going to say it's going to fail whichever way you root it. So there are other alternative splices. So we have alternative splicing. You can splice the zinc finger, alternatively splice the zinc finger transcript and make a different zinc finger protein from the same gene. And so there are ways you can alternatively splice. You can make an alternatively, basically a slightly different part from the same gene. Several parts from the same gene. That's what's great about alternative splicing. So there's a lot of ingenuity in eukaryotic, especially human genes, just amazing primate genes. So there's kind of like, if you can see this, I'll say it's like a Lego-like modularity. So if you take these little boxes, I'll just say that's kind of like a Lego, for lack of a better word. And so I'll say there's a Lego like modularity. It's exploitable. From Wikipedia, proteins that are transcription factors. I'm sorry about my typo there. Proteins that are transcription factors are modular. The first man-made drug inducible repressor that worked in mammalian cells was crab domain, was extracted and joined to a tetracycline repressor domain by engineering. Man-made designer transcription factors are made with designer zinc finger domains and other domains. Synthetic biology is a design-friendly enterprise. Probabilities of evolving functional proteins become better understood through the experience with nanoengineering of proteins. So where was I headed with that? As we try to engineer, trying to make gizmos like this and take this piece and connect it with another Lego piece, we can make new interesting proteins. And that's, if I may get on my soapbox briefly, and I promise I'm not going to make my usual mad rants. If I can get on my soapbox a little bit. If we're taking pieces of genes and splicing them with other pieces of genes to make brand new proteins, that's a process of intelligence. What evolutionary biologists call that is promiscuity, random promiscuity. And you see, sometimes the language, the labels we attach to this, promiscuity is such a derogatory term. If we say clever engineering or clever transplantation rather than promiscuity, you could see that this changes the perception. Using derogatory terms for God's creation affects the public perception. We could use something that is not quite so theologically heavy as saying God's creation. We could say the sophisticated architectural transplantation, sophisticated architectural transplantation of domains. And in fact, the word sophisticated, artful, clever, crafty are terms we're starting to see in structural biology, molecular biology to describe this, cell biology. Those are better terms. It's from the evolutionary quarters we get very negative derogatory terms like promiscuity and junk. Right. So, okay, it's kind of my little soapbox mini rant there. But as we've made these, engineered these new proteins by taking one piece of a protein here and connecting it to another piece of a protein here, and you do that several times, we're able to craft some very interesting pharmaceutical or biotech applications. That is not a random design. That is design with a goal in mind. If this is done by random translocation, you don't know what you're going to get. It could be a disaster for all you know. If you add that crab repressor, just grabbing all sorts of things randomly, that may not be good. Okay? Promiscuity is perfectly accurate. Okay. Thank you for the comment, Immutable. It may be accurate, but it contains undesirable cultural connotations. Like I said, I wanted to have a thumbnail to highlight promiscuity among protein domains. And when I Googled for an appropriate image, I couldn't get one that was family friendly. It's like, you never know what you'll get. Sometimes you get some interesting thumbnails. So let's go on. Crab zinc finger proteins can function as transcription factors and components of chromatin modifying complexes. And so here's an example of promiscuous domains. Okay, so I'm using, forgive me here, I'm going to use the word promiscuous. Promiscuous. So you could see here you can take this crab domain, it can be gone, you can get a scan domain, or this Duff 3669 domain, or you could get the crab B here. You could get the crab B here, and it's there, and then in this protein it's not there, the scan. And you could see all these little pieces are mixed, are mixed and matched. They're mixed and matched in eukaryotic proteins. If we go to the CDART NIH database, you'll really see it. It's really cool, where you have a lot of this mixing and matching. These, if we don't say promiscuous, how about translocated? Translocated is a nice neutral word. So maybe I'll try to, these translocated domains, or these reappearing motifs, or reappearing domains. You'll see them a piece here. You'll see all these proteins that they look like they're just little Legos that were of certain classes and then just stitched together. This causes a problem for phylogenetics. How did these proteins evolve? Where you have one piece just in the right location to do its job, to make the protein. If you take, like, for example, this, let me move it here, see if I can. If you take, for example, here, and you put the zinc fingers, you take the crab A here and put it on the other end here, what we call the C-terminal end. I call it the right side. That's not exactly right. Well, the formal term is C-terminal domain. If you translocate, or like say you take the scan and this crab A here, and you translocate it to the C-terminal domain, that thing's not going to work. Or worse yet, if you take little zinc fingers and translocate them in between, that's not going to work. That's not going to work. And someone asked, what's the functional difference? In some cases, we don't have all the data on what it does. This is actually an outstanding problem in molecular biology. The cell is so complex, we can at best sequence them, we can identify the domains, but we don't know how they have folded. So the folding, this is a good question in terms of highlighting how much we don't know. So we can take the gene, we can do a gene prediction and say it'll make a protein that looks like that. If we're really lucky and we have enough money, a lab, they can actually say, okay, we have discovered this gene is expressed, it makes this protein from this alternative splice, we have identified it, and that's sometimes as far as a lot of labs are able to go. To identify what it binds to in some contexts is harder, especially if has a lot of transient, if you need other factors to help it to bind, particularly transcription factors. And then what cell type are you going to find this? There are cells, sometimes they'll say there are 300 some canonical cell types, 200, whatever the number is. Others will say there's thousands. And then there are varieties of like little, various differences even among these kind of canonical cell types, and then also cell phases, and then stages of development. This could be nightmarish trying to identify the function. And there are some proteins that are expressed only for a few hours in the life of the organism. So in the life of a human, there's some proteins that are expressed for only a few hours, which is just mind-boggling. I'm sorry, I don't have the immediate reference to that. It did have to do something with endogenous retroviruses that were connected to the repression of this, or something like that, or repression or expression. So that's good. What's the functional difference between them here, please? We are elucidating them, and I'm going to highlight where I may talk about where we have some examples. The two examples that have been well characterized, it's taken 30 years to have an idea of how, like say, transcription factor 3A works. 30 years, a lot of professional livelihoods based on that, and a lot of man hours devoted, and then also CTCF. Those are just two zinc finger proteins, taking that much work and decades of research, and we still don't know. And I'm going to try to highlight, if my voice holds up, highlight those two zinc finger proteins, because they're really incredible. They're incredibly sophisticated. Some of these others, one would have to spend time looking at each and every one. That being said, how many are we talking about? We have about 20,000, an estimate of 20,000 genes in the human genome. Wikipedia says that between 1 to 3% of the human genome are zinc finger proteins. So if we take, like, say, 1% of 20,000, that's 200. So 3% would be about 600 genes. And even then, I mean, this is going to be quite a lot of work to identify the function. And some of these are alternatively spliced and then also have post-translational modifications. So there are many modes that these things can operate. We've hardly scratched the surface. And the CTCF zinc finger has post-translational modifications. It's very sophisticated. It operates in so many different modes. To say what function it has, it's more likely to say what functions, plural, because it has like 80,000 different interactions. I mean, that's just mind-boggling that it could have that many functional roles. 80,000? I've heard numbers in the 80,000s. If I'm wrong, guys, please forgive me, but I think where I got that number is they located 80,000 places on the genome where CTCF will bind and have a slightly different role, and you'll see it. The good stuff's coming. So thank you for the great question. I'm sorry if I have to skip some of the questions here and move on, because otherwise it's going to take all night. I'm grateful for the questions, because that shows interest, because otherwise I'd be afraid I'm boring you all to tears. So let's just move on. It could be crab modifying complexes. So if we take this diagram here and just expand it, we could see other proteins. And here's an example of the PDZ domain. This little, like a Pac-Man here, for those of you who knew the old video game called Pac-Man. You could see how there are various proteins, and these are the names of the proteins like PSD95, CO1, GRIP, INA-D. I just wanted to say it's really kind of funny how they name proteins versus organisms. You know, organisms have these big Latin names and just very, it sounds so, it has such gravitas to it. But protein names are just kind of these little almost abbreviated acronyms. That's just kind of the tradition of how it's done. And so you could see here all these translocated domains. You know what? Maybe I'll use the term promiscuous. You could see all these little promiscuous domains here, how they're just repartnering with different partners. That just sounds more scandalous. Okay, so we can just depict. So if you go to the CDART databases, you'll start, if you go through professional literature, you'll see lots of diagrams here where they will lay out the architecture of the protein in terms of domains. And they'll have all these little symbols and stuff. It's cute. And I say this reminds me of children with their Legos. And, oh, here it is, the CDART domain. And we don't have time to actually go to the NIH website and play with it, but it does remind me of these little kids playing with their Legos. And so these are pictures that are from peer-reviewed publications that are trying to describe the protein architecture. And let me see if I could find something here. So ZF is for Zinc Finger, and CRAB is an acronym. I think SH3, I call it SHARC. It's really circ or something, homology domain. And you can see like this SH3 in this color. You can make different proteins with the SH3 and this PX domain. And you can have this SH2 in the pro domains. You can have all sorts of things. It's very interesting. So this is really laying out the whole thing with promiscuous domains. So we have all these things with promiscuous domains. And I'm just highlighting here the SH3 and I'm just showing other pictures, just candy for the eyes. So I picked this out from literature. You can see the complexity of the domains. This is a different way of looking at proteins in terms of their functional components. And like many things, I got pulled off the project because a lot of my time was working with Joe DeWeese on the SciCalc project. So I highlighted here where I'd worked on unannounced, is more or less a peer-reviewed press release that our lab, the lab I was working with Joe DeWeese, was doing SciCalc, and I got pulled away. But you can see here with the, see if I could point there, with the domains there, it's hard to, this has to be rather, to me personally, okay, I'm not speaking professionally here, personally, these domains to be located where they are rather than randomly, it has to be under intelligent direction. So if you assume common ancestry and that these proteins kind of pop up, those domains have to be properly located. You don't want to have all these domain pieces to stitch a new protein and they're in the wrong place you could have disasters. You could have disasters. So just imagine you had some gene duplication event, and now you've altered that protein and you're expressing toxic proteins. That would not be good. That would not be good. And so to me, the idea that you could have these promiscuous translocations of entire pieces of genes and stitching it together randomly, very difficult. So this is actually this is a chance to do a timeout. We have tended to describe evolution of eukaryotic proteins in terms of point mutations, gene duplication and point mutations. This shows where you either have very funny convergences having to happen, or you have actual huge translocations happening. Translocations from one gene that could be many, many base pairs, if it's even on the same chromosome, being translocated there, or God forbid, you have a transchromosomal translocation where you take a piece of a gene on one chromosome and then stitch it with another. And how do you not break the gene? So you have to have a gene duplication event, and hopefully you don't make something too toxic when you do these translocations to make this new gene. So that's why the zinc finger, the origin of some of these classes of zinc fingers is very difficult to trace, because it's almost like they just kind of poofed. We could say they may form synapomorphies that can actually define clades, or very close to defining the clade. It's interesting to see. That's kind of more of an Emery-Moyna question. It's like, can we define clades with synapomorphies of specific sets of proteins? Or does it even make sense to even try to define clades? We can group animals by synapomorphy, but maybe it kind of breaks common ancestry. I don't know. We know, like, for example, insulin-regulated metabolism will be a synapomorphy that will define an entire group, because you either have it or you don't. But that's neither here nor there. Let's just keep moving on. And I really do hope my voice holds out. Otherwise, I'm going to have to grab some coffee here to try to soothe my throat. I don't want to get a sore throat. Okay, so going back again to my professor's paper here, evolution of protein domain promiscuity in eukaryotes. This is one reason I believe in common design versus common descent. So I tried to make my case there, and we'll just, again, this is part of another presentation, but it's a good introduction. It's a good introduction to the zinc fingers and its context. And thank you for the kind words here. So, can zinc finger domains in a zinc finger protein be explained by a series of duplications from single ancestral zinc finger? I say no. And I think I'm going to take up this, I'm going to get some hot coffee. I'm going to run upstairs. I'm in the basement. I'm going to run upstairs and get some hot coffee or something hot to drink and move on. So food for thought. I'm going to post something here in the role of zinc fingers and endogenous retroviruses. So let me describe the role of some functions identified specifically for some crab zinc finger proteins. Like I said, we don't know all the roles of them. So we have here this crab zinc finger protein, and this is the DNA here. That's the double helix of DNA, and then we have the histones here making the chromatin. So the zinc finger will attach, just like a finger, it will attach to a specific region of the DNA. And then the crab, so the zinc finger attaches to the DNA. The crab part of the zinc finger will attach to other proteins, like this CAP1, and then CAP1 will attach to MAGE. And then the cap1 has other domains. So these are the names of the domains. RBCC is one domain. So crab kind of connects to the RBCC domain, and mage connects to the other part of it. We have the PhD domain, which I think is plant homology domain here. And we have the bromo domain. It has nothing to do with bromine. It's actually the Hindu god Brahma. They called it bromo domain. And so if you remember kind of this picture here of all the little pieces, just imagine this one also has little domain pieces. And there it has also a sumo domain and a sur domain. And then the sumo domain connects to the protein CHD3, and NERD connects to that, and HDAC. HDAC is histone deacetylase, I think. And then there's also the CETDB protein. If I could just pause here. When I was sitting in biochemistry class, as a graduate level biochemistry class, we were looking at chromatin modification. Here, at that cascade here, you're just like, oh my goodness, it's kind of hard to do without all the parts being in place, because it just doesn't make sense. So I'm avoiding using the word irreducible complexity, but it's generally that idea. You need to have the system will make sense when you have all these parts, and they have to interconnect. And let me show you one interesting interconnection here, and then I'm going to go grab something warm to drink, and I'll try to be quick. This interconnection here, the crab, the section of DNA that the crab zinc finger will often bind to is connect to. The word is bind. You can also use the word connect, but the formal term is bind. It will bind to either transposable elements or ERVs, endogenous retroviruses. So with that, I'm going to grab something something warm to drink. Maybe I can warm some cocoa, and I'll be right back. Thank you for your forbearance. Well, we'll say a prayer for your children. Lord Jesus, please be with Matt's children. Pigs can fly. Heal them and protect them, Lord. Be with all of us here. We have lots of problems on earth, and thank you that I have this privilege to share with my brothers and sisters and others, my neighbors, on your creation. Be with us tonight. In Jesus' name, amen. Okay, so hopefully it can be seen here that the domains have to be located in the right place to connect to other proteins that have their domains in the right place. There's a lot of simultaneous. And again, we could say connect. The word is bind. For a lot of these things, this is where you have binding issues. So when Dr. Michael Bechly was talking about two protein site binding, it's like, well, here's an example where you have several domains that are binding to DNA and then to other proteins, but then these other proteins have domains that have to bind to other proteins. So sometimes to get a functioning complex, you need multiple binding sites, and he was only treating just binding sites of a few nucleotides. So the question of waiting time was okay, it's like, okay, let's just, how many trials can you keep throwing at this from a certain population? Maybe a bacterial population might be a little easier to work with, but mammalian populations of creatures that don't have a lot of reproductive excess, which means like having only 3 or 4 kids per set of parents. I mean, if your models require the females, each female in the population to have like 40,000 kids or millions of them, like in, the question that Dan Grahr brought up, where it was like something like some absurd number like 10 to the 80th to avoid genetic entropy. And then you combine that with issues of waiting time just to develop new proteins. Those numbers aren't going to fly. So what would we appeal to? People like me would appeal to miraculous events, common design. But anyway, I'm just trying to set all of this up. And again, this is actually starting to touch, people have been wondering what the endogenous retroviruses are used for. If they are binding sites, and I saw this for a long time, if they're binding sites to molecular machines, these molecular machines then can regulate other parts of the genome. So we're finding out that these are basically nice parking lots for molecular machines that help do gene regulation. So you could see that the endogenous retrovirus can sit here and it can regulate another part of the genome. Now this is simplified. I'll show you more complex forms of regulation, especially if they're transchromosomal. It'll blow things away. Yeah, here I highlight some of the domains, the protein domains here. And again, I said it looks like Legos to me. So where I got this one here, it's related to this one. I had alluded to it, where there are proteins that are expressed only for a few hours. And so one of those proteins, it says endogenous retroviruses function as gene expression regulatory elements during mammalian pre-implantation embryo development. That may be a case where the ERV helps regulate the expression of that protein for the few hours that it's necessary in the lifetime of a human. If I got some of the details wrong, apologies in advance, but that is just the reference to it. And so these are some of the mechanisms then associated with endogenous retroviruses. Therefore, one can say that this isn't exactly junk. So when I show this picture, I said it's simplified. It might be more meaningful to see here, where you can have, so this line here, this line, this is DNA, and you have spacer DNA, and then you have these little motifs on DNA where the molecular machine can attach. And then it can serve to do all sorts of things, to do gene regulation. So if you think, if one thinks about how we could take the importance of gene regulation, for the most part, it's not exact, it's more subtle, but I'm just going to give the simplification. Every cell that has DNA has pretty much the same DNA that other cells have. There are really some exceptions, okay? So just take that caveat. How is it that the DNA that makes your nervous cells could be fairly identical to the DNA in your bone cells, or your liver cells, or your kidney cells, or your skin cells, or your teeth cells, or any number of these canonical types? Or muscle cells. It's amazing that you can take the same DNA and make something so radically different. Or like, say, reproductive cells, or cells that make up your ability to hear or to smell. It's through all this combinatorial gene regulation. And what you're seeing here in that slide there is all the machinery that's needed to change the gene regulation so that you can express different kinds of cells from the same set of DNA. And what's amazing is you have these cell lines, so that you know, when it makes a skin cell, it makes other kinds of skin cells. There's different qualities of skin cells, and I don't have time to go into chromatin modification, but these machines can, if I could highlight it here, these machines can change the state of the chromatin. It'll make a chemical modification to the histones associated with the DNA, and even some single modifications to one amino acid on a set of histones in the Hox gene cluster will change the quality of the skin, like say on your eyelids, versus the quality of skin on the soles of your feet. It's amazing. This is what all this machinery is for. It's flipping all these switches. And this reminds me of a factory where you see a robot factory where the DNA serves as scaffolds or robot arms, perfectly positioned. And so there's something that you'll have to notice here. How does the machine know to park on that piece of DNA? It's binding motifs. That DNA has to be spelled a certain way, and then the molecular machine, which would be a complex of proteins, will then attach to it. It recognizes it from out of the entire genome. And so this is the role of some of the non-coding DNA, and even coding DNA can serve as locations where these molecular machines can park. What is really cool is when you do have molecular machines that can park on coding DNA, because then it makes that DNA multifunctional. It's really cool. So when Michael Behe's talking about evolving binding sites, yeah, this is really cool. And all of these will require waiting time problems. And I think evolutionary biologists are trivializing the difficulty of doing this. I don't think their accounting is quite complete enough. You know, I'm not trying to. I respect Dr. Dan and the people there that are studying evolutionary biology, but this, okay, just at a personal level, the way they're trying to describe how this evolved is just not detailed enough, and it really glosses over things that I think are very problematic from a statistical standpoint. It really, that's why I'm a creationist. It's too hard to believe this could just, you have some sort of selection gradient that makes this possible. I just don't believe it. I'd be on the evolutionary camp if I thought it were more feasible. Now, excuse me. Now, I'm a young Earth creationist. If I were a hardened old Earth creationist, I'd be like Michael Bechly. I'd really more like Stephen Meyer. He would think that there'd be miraculous events as the species evolved. It wouldn't. We'd have common descent, but then miraculous events along the way. We call those progressive creationists. Now, there are varieties of it. I haven't really studied progressive creationism that much. So I'd say that as a pedagogical, educational model, I would accept trying to describe progressive creationism, even though I'm a young Earth creationist. It's at least a good model to conceive of what miracles would have to take place if we assumed universal common ancestry. So let me try to keep moving on, because we're getting on 2 hours and 18 minutes and still have a lot of slides. So I was saying that this is like a factory process. And now we get into transchromosomal gene regulation, where you have these proteins binding and then helping to regulate or process genes on multiple chromosomes. And you have these things called like transcription factories. Here's like a spectacular example of that, where you have a link RNA that is involved in drawing chromosome 2, chromosome 15, chromosome 17, and the X chromosome into proximity, and all these chemicals and molecules and molecular machines will connect together and start co-expressing transcripts that eventually become proteins. Just remarkable. Or they're switching in the chromatin state of the chromatin. There's probably some sort of computation. It's just incredible. So I've deviated from zinc fingers there, but I'm going to show there's one important zinc finger protein. There's one important zinc finger protein that helps this looping here. So you see this looping? There's an important zinc finger protein that is very critical to this. So as I was trying to point out, look at all these little locations that the DNA has to be the right motif. It has to also attract the right protein there, and then you're able to get this incredible regulatory complex. I just find this, I think it's going to take a bigger leap of faith to say that this evolved by a gradual process for me personally, versus a creator God with great intelligence putting together something so ingenious. So now, hopefully I won't blow up my computer. Let me show a video. Give me a moment here. I'm going to have to set up the video. I'm going to show that one of my favorite zinc finger proteins here, if you'll give me a moment. It is, if I hope I have it. It is the CTCF protein. And I'm going to introduce the CTCF zinc finger protein. Give me a moment to set this up. Thank you for your forbearance, everyone. Maybe it's not going to like that. I don't think it liked it. Let me kill that before it kills me, before it kills my computer. Okay. Guys, if the audio doesn't come through, just let me know. I'm going to try to make sure I set this up correctly. So thank you for your forbearance here. Let me see. Okay, so this is a window. All right, give it a try. Let me know if you hear audio.
In a recent study, we show that when the 2-meter-long human genome folds inside the microscopic nucleus of a cell, it forms 10,000 loops. Each of these loops connects 2 anchor points which are far apart along the chromosome, but which loop back, coming close together as the genome folds. In our new study, we find that loops form by a process called extrusion. Let's take a look at how this operates on a segment of DNA. First, an extrusion complex containing 2 subunits attaches to DNA, forming a small loop in the process. Next, the 2 subunits slide along DNA in opposite directions, making the loop grow bigger and bigger. The extrusion complex looks for a specific motif, a DNA word, which causes a protein called CTCF to bind to the DNA. If a subunit encounters a CTCF motif that is pointing at it, the subunit tends to stop. If the CTCF motif is pointing the other direction, the motif won't be recognized. To the subunit, the motif looks as though it is backwards. This behavior leads to a result we call the convergent rule. The pair of CTCF motifs at the ends of a loop must be pointing towards one another on the DNA. Our simulations also showed that when loops form by extrusion, they lead naturally to the formation of what we call loop domains. This means that if 2 pieces of the genome are inside the same loop, they tend to bump into each other more often. In this physical simulation, you can see how the extrusion of 2 loops leads to the formation of 2 spatially segregated domains. Like the convergent rule, the association between loops and domains was observed in our earlier study, but at the time we did not understand why this was the case. Both findings, and many others that you can read about in our paper, can be explained by the extrusion mechanism. Extrusion is an exciting model for several reasons. First, loops that are formed by extrusion will be unknotted, so if you need access to the genetic information they contain, it's no problem. You can just stretch them out. Second, extrusion doesn't just lead to segregation of domains within chromosomes. A collection of chromosomes that are all jumbled up will naturally become segregated if extrusion takes place. The fact that chromosomes are segregated in this way inside the nucleus, forming chromosome territories, is well known. Similarly, when several chromosomes are close together, loop formation by extrusion won't lead to entanglements. In contrast, loops forming by diffusion would routinely lead to entanglements. This is important because when cells divide, their chromosomes must come apart. At scales much larger than the double helix, the structure of the human genome is poorly understood. Loop extrusion provides us with a richer understanding of the genome's 3-dimensional architecture.
Praise God for his genius. That's a Rube Goldberg machine that just highlights the genius of our creator. I tell you what, when I was sitting in graduate level biochemistry class, I was just eating this up, and I wanted to just say hallelujah in the middle of my chair. I couldn't do that. It was a very worshipful experience to see the genius that could get something like that to work when it should fall apart. So if you've ever seen Rube Goldberg machines, it's like, boy, that's incredible that it could still work. It's such at the edge of chaos, and it still achieves something. That's what I was seeing in that. So when you saw the different colors in that, it looked like a ball of yarn and the different colors of yarn. those are your chromosomes when they kind of get spread out. You know, they're not compact. So those chromosomes where you see, we have all the chromosomes identified, and they look like all these nice little blotches. That's only in one phase of the cell. The other phases of the cell, when it's kind of doing most of its business, all of that has to get kind of like decompacted, and then it interacts like that. It looks like that ball of yarn. Every cell type has a different configuration of those balls of yarn, and each of those different colors yarn have all these CTCF binding sites. That means all over the DNA are all these thousands of positions that have a certain message on them and say, it's like an address. It's like an address, and the address has to be oriented in the right way, and that zinc finger protein will find it. So how big is the CTCF? See if I have it on my slideshow here. If you'll indulge me here, let me see if I can. I'm glad. Oh, Patrick Alexander. This is great comment by Patrick. Yes. And I think this is only scratching the surface of the wonders of the cell. And, you know, if an evolution, you know, this is, I just want to, you know, I look at some of these evolutionary biologists, I said, your phylogenetic trees are not going to solve the evolution of this level of complexity. Just, I don't see it. I don't see it. I sooner believe in miracles of God than that nature could do this by itself. I just don't see it. It's too hard a step. So let me go back to the screen share here. And thanks for the, Cheryl, it's so good to see you. And Green, good to see you. Thanks for the feedback, Cheryl, telling me that the audio was good. So this complex here represents just one of those loops. And this is the subject of the ENCODE follow-on called the 4D nucleome. There's several ENCODE follow-ons. This is really cool stuff. And then again, also relates to, you put the ERVs, you have all these machines. So what you didn't see, what wasn't shown in that video, they were just highlighting the DNA. But imagine that ball of yarn there that you're seeing in that extrusion loop video, that ball of yarn. That's a simplification, because with those yarns are attached all of these proteins and machines. So you have that ball of yarn moving around with machines attached to them, regulating simultaneously. And it's not just moving. While that ball of yarn is moving around, you have all these machines scurrying about on that ball of yarn, of different colored yarn, scurrying about and doing their thing simultaneously. All this level of activity and coordinated orchestration, incredible symphony. Praise the Lord. It says in the Psalms many times, I will declare the works of the Lord. So for those of you who have suffered through this, hopefully you'll see the point, some of the point I was trying to make. This is glorious. This is incredible. You know, if we just dump all the terminology, that's why it's great to have these graphics. You could see, like, how could anyone make that unless they were God? Only God could make something like that. Humans can't make anything that complex. If we could solve it and alter it, we would have cured all diseases by now. We would have cured all diseases by now. So anyway, let's see if I can, this is so there's different colored yarns. Sometimes they'll segregate, and there's all this. This is a great illustration of all that's going on just in the nucleus of the cell. On top of that, you have, what, 100 trillion cells, and they have to coordinate amongst each other. So this orchestration of all these events from the very small chemical levels all the way to the, the entire full-grown human being. It's just incredible that it's coordinating this. So even in the process of thinking and hearing, there's all this gene expression going on that's helping to facilitate that, and all of the DNA moving and the extrusion loops and all the chemical machinery. So this highlights just all that has to go on and be beautifully orchestrated. So the issue is they're saying, oh, you shouldn't use the word target to describe how long it takes to evolve this one protein. And I'm saying it's way more complex than that. One, it's not just evolving one protein. It could be evolving many other proteins simultaneously with it. And we won't use the word target. We'll just say, what's the difficulty of creating proteins that are that well coordinated? And I showed the picture of the crab zinc finger connected to the MAGE and the CAP1 complex with all those other domains. It's usually not going to be one binding site that you're going to be having to evolve a functional protein. So we're beginning to appreciate the difficulty of actually evolving something complex. Now, the following part with the zinc finger, and I'll go to the CTCF hopefully before I end this stream. And I think we still have at least an hour to go. I'm sorry. So for those of you who missed part of it, there's going to be more. So especially for those on the West Coast. This metaphor here I'm going to present may not be that good, and I might have to rethink it. But consider navigating molecular machines to the right location on the genome is somewhat like doing a text search powered by winds of Brownian motion. So if you take a key and we visit random houses in a city until you open a lock. So let's say you had a key. Let's say you had a key and you wanted to find out which lock matches it in a city. How would you do that? How would you do that? If you didn't know, if someone didn't give you the address of where that key is going to fit, which lock, which house it belongs to. You could go to each and every house with that key. It'd take a long time, and you're going to, okay, forget the fact that that would look kind of weird, okay? Someone would think you're a burglar. But let's just say hypothetically, just a hypothetical example. And you're taking that key and you're going to try to find where it is, where that key matches the lock. So if you look at the problem here of having a molecular machine and it needs to find that right position of DNA. So you have, let's say, a zinc finger protein that's going to find a specific location on the DNA. How's it going to find it? It actually does, it literally does almost do that. It's going to bump around the entire genome until it finds it, and has to do it fast enough. It can do that because the physical 3D space is probably small enough that it can explore it. This is a really tough, I mean, just think about doing a search. So like for those of you in the computer field, if you have an unindexed file and you're going to do a text search, you literally have to go through every location, and you're going to find, like, when you do a find function, like say for a book and you want to look for a particular name or word, the computer will go to every location and highlight that. So this is kind of the same thing when you have a molecular machine that's a protein and it has to find the right location in DNA, it has to do it fast enough. It's just amazing that, so you have all these proteins just floating around, colliding, trying to find their partners. It could be DNA, or it could be like, as you saw with the crab zinc finger CAP1 complex with all those other parts. These proteins are just bumping around, and they're finding each other, and then they're going to self-assemble into a working machine. Fabulous. And Ray says, I wonder if David knew the half of it when he wrote Psalm 139:14. I praise you because I'm fearfully and wonderfully made. Your works are wonderful. I know that full well. Good question, but for us here in the 21st century, we really we have the opportunity to learn it. And it's sometimes, it's sometimes a little demoralizing that we can get called into red herrings when we're talking about Linsky's experiments. I mean, how much print and attention has been given by creationists to the Linsky experiment and to Tiktaalik and to the dino-bird transition? Like, look what you're missing. There is stuff in molecular biology that's just mind-blowing. I look at this and my jaw was dropping, and it meant a lot to me that Patrick Alexander is like, wow, this is amazing. It's like, yeah, that's exactly it. We're not doing just anti-evolutionism here. We are studying and declaring the works of God. And I've gotten feedback from some Christians. They said, yeah, it felt very worshipful when I was teaching the complexity. It's like, yeah, that's how I feel too. I talked to John Sanford when I said, hey, look, John, look what I discovered. And he said, he said to me, doesn't that make you want to just kneel down and worship? I said, yeah, only God could make this. The wondrous works. We're fearfully and wonderfully made. And speaking of which, I mentioned the name of my boss here. So there's also, I say here, balancing between affinity and speed in target DNA search by zinc finger proteins via modulation of dynamic conformational ensemble. Okay, there's a peer-reviewed paper that talked about being able to get enough of the search speed. And I don't know if Immutable, if you're still here, this is an interesting problem for a zinc finger protein. And so this is for the protein chemists out there and the chemists. When molecules interact, they have a certain strength of, I'm going to botch this up because it's been a while since I've studied this. They call it affinity. When, how hard is it to pull them apart? So they'll have they won't have a covalent bond. They'll just interact, be in proximity for a while, and then they could be pulled apart. If the affinity is too strong, they'll stick like super glue. So let's say you try to insert that key into a lock and it just sticks. It doesn't open the lock, but you can't pull it out. It just sticks because it's like super glue. That's bad. What you want to have is that it'll attract just enough, and then it's not too much. If it doesn't match, it'll just release and move on to the next until it finds a stronger match. This is actually pretty difficult biophysics problem. It takes a lot of good foresight to get that balancing. So that's one of the first problems here. Trying to evolve a binding site, it's not just that it will find the address, but it has to have certain properties that it doesn't attach to things that it shouldn't attach to too strongly. And that can be a problem. I'm just alluding to this right now because to really describe this well is like another hour talk. Ironically, Goethe's famous novel on promiscuity is titled Selective Affinities. Yes, okay, so the word promiscuity can be used for the domains, but then it can also talk about the connection, the affinity. So when we have promiscuous enzymes or promiscuous proteins, that means that they can connect to a variety of partners. They can connect. And the strength of affinity is actually very important, because if the affinity is too strong, a lot of these molecular machines will spontaneously assemble at a location in the cell. And when they're done doing their task, they have to disassemble, and then they'll disassemble, and sometimes the parts are used to signal other machines. So off the top of my head, we have like the hot air link RNA, and the complex, when it creates the polycomb repression complex, this machine will spontaneously assemble in the Hox gene cluster. And when it's done doing its thing, it disassembles, and the parts will go off and signal maybe other parts of the cell that this has been disassembled, move on, do something else, or the protein gets degraded. I don't know that, just for example. I'm trying to think if that actually, yeah, there may, if that's not exactly the case, there's probably one system we can identify where once the part, I've seen cascade diagrams, once that complex of proteins is done interacting with each other, it breaks apart the pieces that are now modified. They have like modifications on them where their switches are flipped on or off. They'll move on to other parts of the cell and say, hey, I just finished completing this task. I'm here to do this task and help participate in it. And so all this is being done with Brownian, a lot of Brownian, for lack of better word, Brownian motion. It's random molecular motion. I'm just using the word Brownian. It's just sailing along, bumping around, and it's able to figure out in this super randomness, when it finds the right partner, it'll connect. And then when the complex is connected, it will then perform a task and then spontaneously disassemble. So going back to this thing about affinity, if the affinity is too strong, they're just going to stick together and not be able to break apart. It has to be carefully engineered that it doesn't stick so much. So this binding site engineering needs to be very fine-tuned. And I cited a paper that talks about some of the problems if you don't fine-tune it. It could be a disaster. So thank you for that insight. Matt's, I got to say about the gang here that is on the channel for some of my presentations, the regulars here, you guys are really well read. I can't believe some of the things you guys read. I tend to just focus on a few books, but you guys have just such a large depth of literature. I'm just totally amazed. So I've never read any of Goethe's work. So hats off to you. So going back to our topic, DNA may assist navigation of machines by channeling winds of Brownian motion. Again, Brownian motion in a figurative sense, not absolutely literal. Tuned vibrations by DNA, by resonant DNA vibrations. Let me see if I should do this. I hope I don't blow up my computer or cause the stream to die. Let me just say this. So Lorianne Dent, John Sanford speculated in resonant vibrations. They thought it created radio waves. So my first job was to see if this, one of my first jobs with Dr. Sanford was to analyze the hypothesis that DNA could generate electromagnetic waves. And I concluded using Stokes' laws. There's something called Stokes' law. It had to do with there are many things called Stokes' law, but this had to do with electromagnetic wavelength or something. And I said the DNA is too short. It's not going to create the right wavelengths. We're going to have problems. And that's why Lorianne Dent, who put forward that, she and John Sanford put forward that hypothesis. She had done some experiments trying to detect electromagnetic wave in among the DNA and couldn't detect it. And I said that's consistent with what I would expect as an electrical engineer. So Ben Rex 777, if you're listening, here's a little electrical engineering. That we know antennas have to be a certain size to be really efficient. If it's less than the wavelength, you get you get problems. It gets increasingly more difficult. It's, at some point, just like forget it. It's not going to make an electromagnetic emission. But they did have the idea that DNA had certain resonant vibrations. That's just like when you take a guitar string and you strum it, it'll vibrate at certain frequencies. It is important for navigating transcription factors around the cell. So let me show you something. I know this is going to be off topic, but since we're talking about fearfully and wonderfully made, let me see if I could find this, guys. Point, and this was point-to-point navigation. Yep, that's it. Creation, evolution headlines. I wrote an article on this, and I cited research by certain protein researchers on this resonant phenomenon. So this involves making the zinc finger proteins even more efficient. Let's see, info. Okay, so I wrote this article. On creation evolution headlines. Let me see if I can dump the link into the chat here. In the meantime, I'll just take a little break here. Look at what Ray said. The Miller-Urey experiments run into affinity problems right away, forming melanoids out of the products produced. I didn't know that. The other problem is they used the wrong mixture of gases, making it all moot. Thank you. Yes, affinity is really important. So I'm going to show the highlights of that article. In this article, I did talk about exactly what I was describing. I said, think about pinpoint navigation and propulsion in a seemingly random soup, and the importance of non-coding DNA in helping that navigation. So I say, think of the difficulty of sending a package from one location to another when there are a billion possible destinations. To make this process efficient, it requires infrastructure and machines that can propel themselves and navigate. It's even more amazing if this happens in an ever-changing soup of molecules within a cell. In 2007, John Wren discovered a link RNA, that's a long non-coding RNA, transcribed from DNA on human chromosome 12 that would somehow navigate and land at a specific location on human chromosome 2 by writing a molecular bus known as SUS12 protein, known as the SUS12 protein. It was the first example of a transcript from one chromosome influencing the expression of the gene on another chromosome. This epigenetic action, he found, was a crucial part of cell signaling for differentiating skin cells in the body. It's why the skin cells in the sole of the foot, for instance, have different qualities than skin cells in the lid of the eye. Amazingly, Wren began his research suspecting all he would come up was rather than a real discovery. He thus named this amazing RNA molecule hot air. When his research was published, the journal editors hailed it as the greatest article in their history, but the name hot air was retained. It stands for Hox transcript antisense RNA. Again, this whole tendency to kind of use derogatory terms for God's creation, he called this gene hot air. Anyway, how this feat of navigation and propulsion by the SUS12 protein that shuttles the hot air link RNA from a specific location on chromosome 12 to a specific location on chromosome 2 could be accomplished in a seemingly random soup of chemicals boggles the mind. How this could be accomplished, it boggles the mind. After all, there are over 1 billion locations on in the nucleus to park hot air on a particular stretch of DNA. So the DNA has, like in humans, like 3 billion bases, you're going to find one stretch of DNA that's probably, if I recall, on the order of 10 bases. You're going to find 10 bases out of a stretch of, out of a collection of 3.3 billion bases. You're going to find a stretch, a sequence in all of that that's about 10 nucleotides. Amazing. And you don't have active propulsion. All you have is random, quote unquote, Brownian motion. Amazing. How does this happen? After all, there are only a billion locations in the nucleus to park. There are over a billion locations in the nucleus to park hot air on a particular stretch of DNA, not to mention the additional challenge that DNA is a moving target. So you saw the moving target with the DNA as it looks kind of alive when you saw that ball of yarn. That ball of yarn is moving. What Rin's research demonstrated is that many parts of DNA formerly thought to be junk could serve as navigational markers, like street signs, as well as highways that allow molecules to sail the winds of Brownian motion and deliver molecular packages with pinpoint accuracy. It intuitively suggests a very well-conceived system of highways and shuttles where DNA plays a role. We tend to think of DNA solely as a blueprint, but DNA could also serve multiple roles as a means of navigation and propulsion. The hypothesis that DNA serves multiple roles is furthered by a recent related development regarding proteins known as transcription factors. According to physics.org, rattling DNA hustles transcribers to targets. Let me read this, and maybe I'll show a video here. New simulations of DNA as a transport conduit could shatter the way scientists have thought about how large molecules called transcription factors, and zinc fingers are often transcription factors, about how large molecules called transcription factors diffuse on their way to carry out genetic missions, according to a study by researchers at the Georgia Institute of Technology. The simulations add important brushstrokes to our picture of elusive inner mechanisms of cells. The simulations strongly support the hypothesis that in a live cell, DNA is in constant motion, making it the dominant mover of transcription factors to their target sites on DNA. Yeah, so even non-creationists will use the word target, despite the protestations of evolutionary biologists, to their target sites on DNA. There, the factors regulate the transcription of genetic code into life-sustaining action. So let me see if I could show how this works. Okay, rattling DNA hustles transcribers to targets. Let's see if I can. Oh. It's supposed to move. Oh, man. There used to be a way, this used to be animated. What did they do? They disabled the animation. Bummer. Okay, so just try to imagine that DNA vibrating like a guitar string. Those vibrations, let me turn this back on. So you have the transcription factor here. Those vibrations will move the transcription factor to various locations, and it's going to find the right spot in a sea of a billion different addresses. It's going to move it to the right spot. Pinpoint navigation. You have a billion different places this thing could end up. It's going to find that right stretch of DNA, maybe 10 bases long, 10, 20, some specific. Yeah, so John, you're in our thoughts and prayers. I'll just say a prayer right here. Dear Lord, thank you for our brother, John Maddox. Thank you that he's still alive through this car accident. Please take care of him. In Jesus' name, amen. So, to me, this is really cool. And some of these transcription factors here, like this little ball here, that would be like a zinc finger protein. That's partly how the zinc finger protein, it's optimized to find where it needs to go, but even then it has to have the right matching address. So this is a great process where the zinc finger has, let me see if I can, okay, I'm just going to speak metaphorically here. The zinc finger says, I have to get to that address. I don't know where it is. This is the address I'm looking for. And somehow all the other DNA is vibrating and saying, okay, we're going to shepherd you to that right region where you need to go. And all this random vibration, going to shepherd you to the right location, kind of in the right proximity. And then you just kind of meander around and you're going to find that. And when you do, you just connect. Not really, that is so cool. That is so cool. Ray, just to reassure you, we found out because John showed up on my channel recently. I had a show, I think a couple nights ago, and he said he's just resting. So he was at least well enough to listen in and put a few messages in the chat. So we lift him into the Lord's hands. I'm just going to take a little break here since he's a friend and we're wishing him well. We all know his story, many of us, that he had that brain hemorrhage and he should have died. And God could have ended his life, but chose not to. That turned around John Maddox's life. He said he'd commit his life to God after 10 years of just doing his own thing, and 10 years to the day almost was when he had that brain hemorrhage that should have ended his life or made him permanently paralyzed, and he was spared. So I'm trusting God that John is in good hands. So continuing on, and I'm sorry they didn't have that. I'm sorry they didn't have that animation going anymore. It was really cool to see that rattling DNA. So going back to the presentation here. So I talked about, oh, wait, that's the wrong presentation. Hang on. I'm going to kill this one. Let me just kill this one here. So I mentioned Lorianne Dent and John Sanford. They speculated that DNA, the sequence helps it achieve certain vibrations. They thought it was generating radio waves. My first job was to analyze that, and I said, no, that was a gallant hypothesis, but I think it's wrong. But the tune vibration, the acoustic vibrations have actually a role, and that was what I just covered. And so they've been vindicated. They've been partially vindicated. They at least foresaw it. They said this, not only does, DNA is optimized to vibrate a certain way, and you have all the other things that will just help it to navigate these zinc finger proteins all inside the cell. Now, so going back to our metaphor where we're trying to get the key to fit the right lock. So if we can design a protein with the ability to target a specific DNA quote-unquote address, Brownian motion, quote-unquote Brownian motion, and fast random molecular search helps take care of the rest. Doesn't solve all the problems, but it's a big first step. The address is called DNA binding motif in the literature. It's called just a binding motif. So we can have an array of multiple side-by-side zinc fingers to target a specific DNA address. This isn't a hard and fast rule, but one zinc finger targets approximately 3 DNA bases. And there are a lot of caveats to this. So please forgive me if you see deviations from this. And one of them is the transcription factor 3A. And see if I have a picture of transcription factor 3A. I don't have one handy. Apologies. I wish. That's an oversight on my part. But you can have, like, transcription factor 3A has several zinc fingers like that, and it's going to target a specific segment of DNA. So that DNA would be spelled like something like this. And this is actually this is a segment of the 5S DNA that codes for the 5S RNA. And so you'd have to engineer each of these zinc fingers to connect to a certain motif. Now, I hope you're starting to see the problem is, like, if you take, like, say, see if I could highlight this. Say you have a zinc finger that is targeting AAC. You want to have one that will target beside it BCG, and then so on and so forth. And when you string them together, you can identify a specific segment of DNA. If we have what we call a monovalent zinc finger, that's a term I kind of coined. It means it targets a very specific DNA sequence. This is how you would do it. Now, human engineers can only get 3 of them to work at a time. We've never been able, we have a very hard time making them above 3 because there are other complications. Our biophysics is not sophisticated enough to rival what God has made yet in terms of being able to engineer zinc fingers that could target something this long, a stretch of DNA this long. I think the transcription factor can actually identify specific DNA or RNA sequence that's around 50 bases. It's huge, just amazing. And this also ties to the reasons that it will identify or single out DNA of a certain length. Because it has to have the right affinity. You don't want this thing floating around and maybe only a part of it will bind to some piece of DNA it shouldn't have bound to. You want it to not have affinity to other parts of DNA. This is a tough engineering problem. You either change the zinc finger or you change the DNA sequences globally, because if they're DNA sequences that let me highlight this again. If you have DNA sequences here, like let's say you have these 3 zinc fingers, and there happen to be many places that have these sequences here, like you have like that. If there are other places on the genome that have those sequences, that would be bad, and it attaches there, or worse, that it attaches to, like, segments like even that short. It has to be just right. It's kind of a Goldilocks zone. Just pointing that out. This is not as trivial as, I'm sorry to rag on the evolutionary thought process, but do they even think at this level of detail? Do they? I mean, really? Do they have anything? Do they have anything more than just making phylogenetic diagrams and saying this evolved? Do they ever actually think about these careful details of how much can go wrong? And therefore it would be selectively disadvantaged, and then it'd be thrown away. So you might as well just do a random search, because you're not going to build this incrementally. That's what I was trying to go, where I'm trying to go. You're not going to build this incrementally. We also know this from experience, because the engineers have to actually think about how to build this. They have to build algorithms and computers, and then even then it's not so good. A random search is going to have a hard time arriving at this, and what would be a disaster is if you start to have just, like, say, a protein that has 2 zinc fingers. It's going to bind all over the place on the DNA, and who knows, you could gunk up the DNA. You have molecular machines parked in the wrong parking spot. It's going to be a disaster. Have they ever run simulations on this and thought about it methodically, like we're doing here on an internet YouTube channel? They don't do stuff like that. I have problems with that. It's like, guys, can you at least do a little more thought than taking sequences and throwing it through a phylogenetic reconstruction software and then declaring you solved the problem? I don't see them go to this level of detail. But I, because of work like Michael Bechly, where he actually studies this, and he says it's difficult. It's like, yeah, I wanted to follow on with that. That's why I like the zinc finger, because he was in Edge of Evolution. He was studying this in the framework of malaria and chloroquine resistance or something like that. I think the stuff with the zinc fingers is way cooler. So this is a bonus. This is a bonus. You get to see the wonder of how some of this works in the cell. That's why I had more fun with this. It has Edge of Evolution type arguments in it, but then you also get to see the complexity of biology and the miracles of God and the wonders of his works. The genius. So let me go on here. So like I said, if you can imagine, if you have these zinc fingers here, just kind of like metaphorically, you can have the zinc fingers, and I'm highlighting it here. So you see, like, where you have these little, like kind of like omega or horseshoe symbols. And I'm just kind of associating where, like, if you have a horseshoe there, it may correspond to one line here, etc., etc. Do you see that? It's kind of hard to draw this. You can also metaphorically say it's like a key. You want to call it a lock, that's fine too. I just don't have, some of the metaphors are kind of hard to come up with. And so, just like with keys and locks, they may obey the same general shape but have slight differences in the notches and slots. So the zinc finger fold will be about the same for each of these zinc fingers. But the spelling of each zinc finger is a little bit different, and you have to adjust. When you adjust the spelling of the zinc finger here, which I'm highlighting, each of these rows that have the colored amino acids, the slight changes in the spelling enable it to attach to a different sequence of DNA. And then when you concatenate all of these together, they're able to identify a long sequence. So, how does this start to relate to the waiting time problem? Do you see how long, do you see how big that array is? That's a lot. Okay. Look here. Look at how many amino acids it takes to identify a specific section of DNA that is going to navigate that protein. So when you're typing in an address and you put it in the mail and it's going to send that package to the right place, you try to get a lot of the stuff right. You have to at least get the zip code right and then the street address. In a similar way, you have to spell a lot of that protein correctly. A lot of it has to be correct so that it can find a stretch of DNA like that. So we're talking binding sites that are large. Michael Behe had these binding sites, protein binding sites, 10 long. To get the zinc finger, how many is that? That's like 30 times like 100. Maybe you don't need to get you definitely don't have to get all of them right, but you have to get a lot of them right. So 30 times like say 10. So that's about 300 amino acids. You get some of it wrong, it could be a disaster. I mean, we don't exactly know how many. And so the first thing I'd say is, how can you make that assertion that it can incrementally grow? You haven't even modeled how difficult it is. We don't even know how to model it. Once we get beyond about 3 zinc fingers, we have a hard time modeling how well it's going to perform. And so it's really presumptuous that you could just take a phylogenetic tree and say that proves it evolved. That's terrible. That's not being very skeptical or scientific and rigorous with your model. And one thing I wanted to point out, when you change the sequence here, it's going to jump to a different location on the DNA. That may not be what you want. So you have two options. Either you evolve the protein to match the DNA, or you evolve the DNA to match the protein. And one solution that evolutionary biologists have come up with is coevolution. They both evolve to eventually match each other. And I said, did you ever think about how difficult that's going to be? That's what I rhetorically think. You mess up a specific transcription factor, it could have devastating consequences on the organism. Or if that transcription factor doesn't even exist and you have this protein and it's not regulated, how's that going to work? What if you have this zinc finger transcription factor jumping around all the genome in wrong locations and it's not regulating what it needs to regulate? That's bad. I mean, just think if you had a protein that's never activated, it may be in the right sequence, and then the transcription factor, the population's not going to wait around for that transcription factor to eventually evolve. I mean, especially if it's life critical. End of story. So, again, I haven't gone through the rigors of population genetics, but I'd just say on an intuitive level, this is hard. Look at all these amino acids that have to be tuned to get a sequence like that. So roughly speaking, an array, roughly, and again, there are many caveats, but roughly, if you have every zinc finger can identify 3 DNA bases. So if you have 13 zinc fingers, 3 times 13 is what, 39? 39 DNA bases. And so, Ray's question, our resident chemist, I wonder how many permutations of amino acids, 20 to the 150th power. Yeah, this is, once you start to see how much you have to tune to get this work. One reason also that I'm making this little bit of a detour, yeah, and thanks for that comment here, Matt's, yes. Unspecified telic drivers of coevolution. Coevolution. Do these guys even think about it all when they trot out these names for things that they might have happened? So one reason I'm also taking this detour, a lot of the creationists and ID proponents keep citing Doug Axe's work. That's for a different protein, the beta-lactamase. And I'm not trying to be too critical because his work was done about 21 years ago. Now we have stuff like zinc fingers. We can actually, I want you to see how we can estimate the probability of functionality here. I'm actually trying to illustrate. See, now you see kind of because when I read Doug Axe's paper, I was just like, I have a real headache. That was so hard to understand. So I hope I've made this a little easier. You could see, you can kind of conceptualize that if you change this, it's going to change the DNA it connects to. It's that simple. Like, maybe about approximately 3 bases, not too much, not too much. Approximately, it can vary, okay, depending on the transcription factor 3A it violates that rule of 3. It's more like 5 that it can effectively look at. So there are actually programs on the internet, and they're terrible. They don't work very well, but they're about as good as we have it, where you can say, okay, I'm going to type the DNA sequence I'm trying to target, and then it'll try to build a zinc finger for you. And so the reason I know it doesn't work so well is I've taken known targets of zinc fingers, put it in the algorithm, it would totally make a different zinc finger that wouldn't work. And I put it through another algorithm that would say, okay, this is, it'll generate the zinc finger. I'll go to another computer piece of software and say, okay, what DNA do you predict it's going to bind to? And it's a totally different one. So I know as a matter of principle, these things aren't working so well. But you could see now that you have to modify these 32 amino acids to target 3DNA, the right DNA base pair. But in addition to that, not just targeting it, it has to have the right affinity. Not too much, not too little, but just right. And in connection with the other zinc fingers. And Alien Robot Ghost, well, thank you for visiting this late night hour. And we've been going for 3 hours, 9 minutes. My favorite Evo babble is evolutionary stasis, meaning no evolution occurred at all for millions of years. Okay, so now you're seeing that we have other examples where we can understand protein probabilities and binding sites. So that was a big contribution by Michael Bechly, trying to describe protein probabilities in terms of binding site evolution. And zinc fingers are just ideal for studying this. And let me see, Matt says, again, on a purely neo-Darwinist conception of life in which junk is fumbling clumsy toward function, such software should work better. The software failure is a form of evidence. Okay, so where was this? Where did we begin? I was talking about the creationists in peer review, and they were talking about transcription factors. A common transcription factor is the zinc finger protein. And I'm now trying to just show how, with some of the mathematics that they were going through, what they're actually trying to describe. And by the way, I need to emphasize this. They describe systems that involved multiple genes. I'm just describing the difficulty of making one gene. They're describing the difficulty of, like, say, not meaning to put words in their mouth, but like, say, the CAP1 complex I showed earlier, the CAP1 crab zinc finger complex with all those connections, the zinc finger crab protein, one part of it's binding to the endogenous retrovirus, and then the other part of the zinc finger protein, the crab complex, is connecting to the domains on the MAGE and the CAP1 complex and their domains there. The CAP1 complex is then connecting to a whole bunch of other things like the SET1DB. Okay, you get the picture that paper on the waiting time problem is dealing with all these multiple genes simultaneously. I'm showing the difficulty of just evolving this one to find that one lousy stretch of DNA. So when you get down to these details, it's like, these discussions could be easily dismissed by evolutionary biologists. I'm trying to show that the level of detail that they need to go to either make a convincing case or decide, hey, can't evolve, which I would hope they would ever do. But they can stay in that kind of illusionary world that this is evolvable because they built a phylogenetic reconstruction. They can stay there if that's all they do. Because once they start to get to these details, they have to invoke these magical mechanisms we call coevolution, and these things that are harder to believe than magic, personally, because I sooner believe in a miracle than trying to argue that random mutation in natural selection can evolve something this complex in steps. They're more than welcome to describe what the selection pressures actually are and try to make at least one detailed model that'll work and then create an experiment. But right now, all they have, their best play, is just a phylogenetic reconstruction, which isn't really a mechanistic description. And that's where I say there's a double standard. When they debate creationists, they keep demanding mechanisms. And how did this happen? How did this happen? They don't apply that same standard to themselves except to say vague appeals to mutation and selection. But that's not really, that's so vague. It's like, okay, what are the selection pressures? Describe them in steps. What were the mutations that you would say are feasible? They don't do that. They don't apply the same standards to their own models that they keep demanding from creationists. Of course, creationists could be, evolutionists are very good at concealing the weaknesses in their theory, and especially in debates. And this, again, touches on Brandolini's law. Do you see how long we've been going for 3 hours, 13 minutes to this level of detail? There's no way that this could happen in a 1-hour debate on waiting time problems, but this is the level of detail that's needed to really explore the difficulty of what's in question. And that's why I don't like debating this in a live debate. It was great that we had people from the other viewpoints joining early in the stream, but they haven't, unless you guys are here, you haven't waited to hear some of the punchlines 3 hours later. But this is the level of detail. So now I'll show some more diagrams. So we have this zinc finger architecture here. And this will show the zinc finger actually in terms of like the alpha helices. So if we have like the DNA that it's going to bind to, see if I could show this correctly. So let's say we have to, we wanted to get a DNA, and we wanted the target DNA is GCG, TGG, GCG, and then X meaning any other DNA. We'd build 3 zinc fingers, and so one zinc finger would look at GCGT, and the other one would look at TGGG, and then GCG and X. Now, I actually read off 4. So one zinc finger will actually target 4, but then the other one it'll overlap with the other zinc finger that has to, that'll be the first base that's actually the 4th base that this zinc finger ties to. Do you sort of see that? Sorry to be confusing here. The net result is, on average, each zinc finger will target 3 bases. But in actuality, it's more subtle than that. It actually has to target 4, but with the overlap of the 4th in one zinc finger to the first base that the adjacent zinc finger will target. So zinc finger 3 would target these 4 DNA bases, GCGT, In zinc finger number 2, it'll target TGGG. So there's an overlap. It has to be coordinated that one zinc finger and the other zinc finger will target the same DNA base. If you want to have a lot of binding specificity, sometimes that rule is broken, like in CTCF. See, this is getting really complicated. I'm sorry this is starting to get confusing, but there are all these subtleties here you have to think about in terms of the function, because if you're going to evolve this final function, think about all the intermediate steps where this elegant system will not work. Can't just hand wave and say natural selection mutation. That's just not going to cut it as a model of, if you're going to demand detailed models of creationists, the evolutionists have to do the same, and they don't. All they do is make phylogenetic trees and circularly reason that if you can build this tree, that it must have evolved. That's not a proof. That's circular reasoning. Yes. What you said here. But I just wanted to show kind of specifics of the details here. And as you ponder this, now that you know more of how this actually works and connects, you can see some of the evolutionary problems more in detail. And I gave the references here for where you had this DNA engineering. But again, the caveats are these are derived from the models that humans use to engineer, and they're not as good as the one that God makes. The one God makes is way better. So let me see here. So I had an example, like if you had like Federal Express and you get the zip code wrong. Okay, I'm going to show what happens when you change some of the letters of what DNA is targeted. So let's say you have a Federal Express truck and you supply a zip code that's 12192. Zip code 32192 is close sequence-wise to 12192, but it is distant location-wise. So you can see zip code 12192 is up here. I guess that's around New York. And then 32192 is down in Florida. The difference between them, they both have the last 4 digits right, 2192. It's the first digit that is different. It's the first digit that's different. And what does that mean for our discussion here? You're not going to get an almost right zinc finger, and then it's going to geographically go closer and closer to the right location on the genome. You get some of those amino acids off, it's going to jump around to a different location on the genome, if at all. I mean, it may not even bind. It may not even have the right affinity to bind. There's no gradual route to getting closer and closer to the right solution. Because, and I talked to a long-time opponent on this, and he got kind of angry because I've been debating him for 20 years, and he used to kind of, we used to fight each other, and I'd score a draw, but now I'm just bulldozing him. He didn't like that because at the time I didn't have my degrees in physics and biology like I do now. And so I began to learn more, and so my arguments got a little better. And I said, so what's going to happen with these intermediate stages? You're going to have these zinc finger proteins binding to random regions on the DNA and attaching. That's not going to be good. What if it binds to something and prevents another transcription factor from either repressing or activating? That's really bad. Or at best, it just latches onto some random site and maybe degrades genome function a little bit, but you still don't get it to the right location. there's no gradual, like it's getting better and better. I mean, there could be, but on average, it's not. This is not good. So when we're, what I'm trying to illustrate here is when we're tweaking the spelling here, it's like trying to tweak. You're going from 12192 to 32192. It's jumping all over the genome, binding to random locations. Is that good? Did they ever model that, the effect of that? Of course not. Of course not. And so, I mean, I could, I could just say, here you have the FedEx truck as let's just say the FedEx truck is like the evolving zinc finger here, and it's just going to jump all around. It may not be quite the gradual route to the right location in the genome that you're expecting as you're getting random mutations. I think this went over some of my evolutionary biologists colleagues. They really didn't get the problem. I'm just like, okay, guys, I'm moving on. So, I did in my again, this is kind of pieces of a presentation I'd made to some of my ID colleagues. I call it Dr. Sanford and I were thinking about a project where we could talk about the difficulty of evolving proteins. We called it PQuest. That idea never bore fruition because we got pulled onto other things. But just kind of tracking kind of the history of this. In 2004, Doug Axe calculated protein improbabilities stated in terms of enzyme catalytic ability, specifically the beta-lactamase protein. 2017, Michael Bechly, protein improbability stated in terms of proteins connecting to other proteins. So that's edge of evolution. 2016 was the waiting time problem of integrating and fixing functional proteins in a population, but that was from a very abstract level. And in 2019, I was throwing on the table, and this was unpublished, this was totally unpublished work. I was throwing on the table the idea of zinc finger proteins, and that is, I'm working on a project with Dr. Sanford regarding zinc finger proteins, but we're only mentioning the waiting time problem in passing. It's just so cool to talk about zinc finger proteins in isolation without even waiting time, just to declare their complexity. So where that publication will go, at the very least, we're hoping we can get it in peer review. If they say, well, people have already said this, then it'll be an educational paper. So what you've seen in this 3.5-hour presentation so far is actually kind of just showing you parts of our peer-reviewed paper that are just purely educational, just to say to the world, look how cool this is. Look how cool this is. And we're trying to put it in terms people that are non-molecular biologists can kind of appreciate the complexity. So now let me see if I have more. I have more. Okay. So let me look. Give me a moment here, and I showed you the chromatin extrusion loops. I'm gonna try to, and with the CTCF, let me see if I have it here. Yes, sirree, I have it. CTCF. Let me just get this set up. And I know it's been long, but this is You know, sometimes when I put this together, I don't have an opportunity to rehearse this. You can imagine if I rehearsed this presentation 3 or 4 times, that would already be like 2 days worth. So sometimes this is on the fly. This was a lot of work to think about this, and I'm really glad you're forbearing with me, as this is a long presentation, but I just needed to get this out of the way because I haven't done one of these nerd sessions in a while. And I really want to show the YouTube community I'm very serious about trying to make creationist arguments, especially ones other creationists haven't made yet. And just to do a lot of self-promotion here, I'm really eager to maintain that #1 spot as #1 YouTube creationist. That's why I'm making presentations like this. It may just kind of be a little hard on the audience, but I want to say that I'd like to think that the content is substantive. So we saw that video on the extrusion looping and CTCF. I'm going to show you how just amazing this is. So this is the human CTCF sequence. So the name of the protein is CTCF, and that's an acronym for something that's several acronyms. Okay, so like I said, proteins have these abbreviated names. They're usually acronyms. So this is the sequence of CTCF right here. And I'm going to have to decrease the size so you can actually see this a little better. Now let me show the zinc fingers there. See the zinc finger? Now it's not exact, but where you see the colored amino acids is where the zinc ions would connect. So there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There are 10 or 11. I think if I counted that, I don't know if I counted that right. Anyway, so we'll say at least 10. 10 zinc fingers. So those are the zinc fingers. Zinc finger domains. There are other parts of the protein, like targets for post-translational modifications right here. The serines change. This section here is, it's like a connection port, just like your computer has all these little ports where you can connect. This sequence here is where another protein or something else will connect to it, and that's the SH3 domain. So you have you have the zinc finger domains here, you have the SH3 domain. I haven't found any literature that describes the role of this piece here, but still, this is really cool. And we can describe the zinc finger kind of like that. Now, we were talking about those chromatin extrusion loops, and I'll represent it kind of like that. And there's a mistake here that I need to kind of correct in the slides, but just if you could all forbear with me. So this loop, each DNA chromatin loop is about 200,000 base pairs. And I said this is the gene here. That could be wrong, so apologies in advance. In Contested Bones, I actually contributed to that book, even though my name is not acknowledged in that book. I did the parts of the book that dealt with kind of the complex chromatin regulation. I talked about ALU elements, and I emphasized the idea of these regulatory loops. And I'll show you where CTCF plays a role. There is CTCF connects in a certain way to the DNA, and the DNA has to have the spelling in the right orientation and on the right correct strand of DNA. Let me show that a little bit more. So the DNA will have it, like say on one strand, the binding motif there, and then you have the reverse complement on the other part of the DNA. It has to be, so CTCF will bind to the, will bind here, and then it has to bind there. It has to be actually spelled, it has to be spelled in one direction here, and then it has to be spelled backward over here, roughly speaking. So you'll have two CTCF proteins, one will bind here, another will bind here, and that helps the cohesin complex. It works in concert with the cohesin complex to form this loop, those extrusion loops that you saw that were just so cool. So not only do you have to evolve CTCF, you have to evolve the binding sites on DNA to be in the right location to make these loops of the right length, et cetera, to do, and so to do all those amazing Rube Goldberg things. Not only that, you have to spell it in the right direction. You can build all these other kinds of loops. This is just nuts. Maybe I don't have time to go into the complexity of how this has to happen, but wherever you see these cohesin loops here, these little loops, CTCF is lurking nearby, having to help interact with this. So you have to have the DNA spelled in the right locations, those motifs scattered throughout the genome. How's that going to evolve? And I should mention, a lot of those regions happen to be sign elements, these junk DNA elements. That's what they're used for. They're address locations to park CTCF, these molecular machines, to form those loops. So now you see those extrusion loops that I was showing you, and you're thinking, wow, that's really cool. Now you know a little more of the role of the sign elements and these transposable elements that they call junk. It's not junk. And what they say is an evolutionary hierarchy, it's actually a control regulatory hierarchy. And I'm going to show something that's really cool, the Sternberg-Collins paradox. Okay, so let's say let's assume for the sake of argument there's common ancestry. Assume for the sake of argument there is universal common ancestry, but intelligent design along the way. So again, just for the sake of argument here. What did I do? I lost my apologies, everyone. Okay, I got everything set up. This was found out by Richard Sternberg, and he was citing a paper by the NIH director, Francis Collins. And it suggested designed evolution. And the paper was called Discovering Science in the Genome. Richard Sternberg commenting on Francis Collins' paper. So let's say we had a book called, we'll call her Ancestor Heir. And we make two versions of another generation of that book. One we'll call Jane Eyre. Everywhere in the book it says Jane, we'll have one where it says Jane Eyre, or it uses the name Jane everywhere in that book. Let's say hypothetically we have another book, and instead of Jane Eyre, it's Mary Eyre. So everywhere in that book where it says Jane, we replace it with Mary. So we could still say that there's common ancestry, but that's a very deliberate change in locations in the two versions of the book. You'll see where this is headed. So that may sound kind of confusing. Why would anyone do that? Of course, no one would actually do that with the book, but let's just say hypothetically we did that. The Sternberg-Collins paradox points out, see if I have a picture here, that in mice, the sign is spelled this way. It's called the B1, B2, B4 sign in mice. In corresponding locations in the rats, it's called the ID sign. Now, evolutionary biologists superficially didn't think this was a big deal, but what they said is these two bits of DNA were, I mean, this sign had different evolutionary origin than this sign. That it's not like, it's kind of hard to explain this, but can you imagine that all the places where you have Jane in this book, it's spelled Mary in this book, and these are very different locations, thousands of locations in the genome that got reformatted simultaneously. Thousands of locations. This makes no evolutionary sense. And maybe I'll cover that sometime, because that is just, maybe if I can draw pictures of why that is so difficult to evolve. And I think that totally flew over the evolutionary biologist head. The point that Richard Sternberg was making, and by the way, Richard Sternberg is an evolutionary biologist. So just imagine that, how are you going to evolve a book from one book to another like I just described here? You have to evolve all those locations simultaneously to get the effect of going from Jane to Mary. But in DNA spelling, they have a certain kind of transposable element called signs. And the sign is spelled one way in the mouse, a different way in the rat. So even though hypothetically we have the rat and mice descend from a common ancestor, okay, so we'll just assume common ancestry, the rat and the mouse descended from a common ancestor, their genomes got modified simultaneously in multiple thousands of locations simultaneously, just like taking like those two books. The odds of that are just astronomically remote. Why would that happen? Where do you think those CTCF binding sites are? Right there, a lot of times on the signs. Those extrusion loops, so critical for regulation. So you've had to have the DNA modified thousands of locations and the CTCF binding zinc finger to target those locations. You have to have coevolution. I sooner believe in miracles. Okay? So even if you assume common ancestry, you're still going to have to evoke miracles of special creation. And so this is where I think, I hope this is like really cool for you guys, because you're hearing now that ways that junk DNA is used that you may not have been told. I was in a debate with my distinguished colleague, Daniel Stern Cardinal. I didn't have, we're going already almost on, we're approaching 4 hours of talking. And it's like, you can throw these ERVs and signs and transposable elements to get to the level where you can analyze the difficulty of evolving them all and describing their function. It takes a long time. It's very easy to dismiss them as junk until we get to levels like this and actually talk about the difficulties. Because Sternberg actually saw the difficulty. He said, you can say that this is junk, but why is the junk geographically located in corresponding parts of the genome like they just got inserted there? It's like they're optimized to be there. And by the way, they're really, other papers have shown that these are critical to forming these chromatin extrusion loops involved in gene regulation. This is just crazy. crazy, insane design, simultaneous. Even if you assume universal common ancestry, you need miracles to have this level of design. And let me see. So one last thing, let me show some fabulous things about zinc fingers. I had some incomplete work. This ended up not being as good as we were hoping. This is the zinc finger protein of I wish our distinguished YouTube's favorite daughter, or modern day debate's favorite daughter, Erica Gutzik-Gibbon. This is a zinc finger from a Pseudimangabe, a primate. And this is a zinc finger protein. We don't know what it does, but I want to show, look how many zinc finger domains there are. There are 33 zinc finger domains. 33. Okay, I showed you one with a zinc finger 120 whatever, with only 13. This one has 33 zinc finger domains on it. What does this thing do? And this is in a Pseudimangabe. The corresponding human one is here. And I was like, there are enough differences between the human and chimp. I said, the human and Pseudimangabe, it's like, how difficult is that to evolve these zinc fingers? Because you have to simultaneously evolve whatever targets these are. I'm presuming it's DNA. It doesn't necessarily have to be a DNA target. So you either evolve the DNA it targets or the zinc finger, or both, which would be a co-evolutionary scenario. And if that DNA targets on multiple locations on the genome, that even makes the problem worse. This is really funny. I'll tell you what he said, because I showed the Sternberg-Collins paradox on Skeptical Zone. He appeared and he said, oh, that's easy, you just need transposases. And I was really confused, because actually the right thing you need is reverse transcriptases. He didn't even get the enzyme correct. So yeah, you couldn't even get that correct. And then it's like, well, how do you know that the reverse transcriptases are going to insert the DNAs in the right location? He had no explanation. It's somewhere, just for what it's worth, Mats, when you asked. I think I'll drop it in the chat, and you could see the interaction. He didn't say much. He didn't say much. But just for the record, since I claimed I had an interaction with him, and I seem to remember, let's see. And it was Sternberg Collins. Hang on here. I'm going to try to, yes. Somewhere in the comments section, he weighs in and we interacted. I'm going to drop it in there. Oh, yes. So this explains what I just did in even more detail. And you could see what Swami Das had to say. He couldn't even get the right enzyme mentioned right. I mean, he was just saying all this stuff at the top of his head and didn't even name the right enzyme correctly. See, but that's behaving like a typical evolutionist. You'll just stuff off the top of your head. We have a phylogenetic reconstruction. It's taken care of. The problem that Sternberg identified, even assuming you had a phylogenetic reconstruction, the mechanism that can locate it in the same place it's hard to account for. And so, I mean, this was, yeah, so just for the viewer's benefit, this was the beginning post I had. Somewhere in the comments section, there's Swami Das. Yeah. You can even there see Swami Das. People were interacting with Swami Das. And I politely called him out that he got the enzymes wrong that were involved in transposition. I used to be posting on his hostile pseudoscience website, and it just, it was productive for a while, but I moved on. Yeah. Well, the problem is Brandolini's law. I have, it took, the way this debate would work well is if you give Dave Farina 10 minutes and James 2 or 40 hours to take them apart, because that's the problem. It takes so much more effort to refute garbage than it is to generate it, it's easier to make BS a little bit. It's easier to make make BS than to clean it up. Much harder to clean it up. Yes. Yeah, that's very sad. And I got it just as a little bit of an anecdote. When I was walking the halls and sidewalks of the NIH, I kept thinking of Richard Sternberg. At least perhaps one of my professors was his colleague there at the National Center for Biotechnology Information. He might have worked with that group. That might have been the group that fired him, and my professor was part of that group. So yeah, I thought of him a lot when I was taking classes at the NIH. So let me show, well, since we're here, I'll show the signal. We're showing the locations of where the sign occurs in the mouse and the rat. That's what these diagrams are. It'll take a while to uncoil this, but you can read that. And that's where I said, just think about Jane Eyre and Mary Eyre and the problems. You might be able to build a phylogenetic tree, but you still are faced with difficult problems as to why why it is that even on the same corresponding locations in the rat and mouse genome, you have that sign there, differently spelled. And he's very astute. He picked up on that. But it makes more sense from a design perspective where you're having that CTCF protein that's looking for these various signs that happen to contain the correct binding motif and also oriented. It has to either be spelled one direction or reverse spelled the other direction for the binding motif to work and to build all those extrusion loops in the right way, because otherwise you'll have a mess. So one way to think about it is just like you have a parentheses one way and then you have a parentheses the other way. To make those loops, you have to have DNA that's kind of like a left parentheses in one location, and then 200,000 base pairs later, you have one in the other direction. How does evolutionary biology explain these kind of nicely paired punctuations that are just the right distance and the right orientation, like a left parentheses and a right parentheses, or vice versa? So not only are these paired, but like in the case of the rat and mice, the binding motifs are different, which means you need different I'm sorry, I'm looking at the side chat here. You guys have a great sense of humor. Not only different binding motifs, you need to have the corresponding change in the CTCF. How does that evolve? You can invoke coevolution, but like I said, when they start talking like that, it's like you might as well start invoking miracles. So let me see if I could finally so what I tried to show here with the Sudi Mangabe and human, it's going to be tough to be co-evolving these things. You have to change the DNA it's targeting simultaneously. That's not so easy. But as we looked at this, we said, yeah, it looks difficult, but to actually make an argument that it's improbable is going to be hard, and to apply the waiting time to it. I said we don't know enough about the zinc finger biophysics yet to really make a killer argument. So right now it's just a good intuitive argument for waiting time, but it does show the wonders of creation, just the ingenuity of the systems that these zinc fingers are involved in. So zinc fingers will make transcription factors, but then one zinc finger in particular, the CTCF, helps make these extrusion loops. So before I wrap up, let me cover two things that I promised. I was going to cover and hopefully I could do this in 10-15 minutes, maybe even less, the transcription factor 3A and the CTCF. So I'm going to, hopefully my computer doesn't blow up when I'm trying to retrieve this. Give me a moment here, guys. If you'll forbear with me. Microsoft Word will take a while to grab this. Okay, I'm just going to read from something I'm writing here. So this is an unpublished work. It's a draft. So let me just show it here. CTCF is a zinc finger protein that can bind to 80,000 locations. It can bind to 80,000 locations in the human genome that have varying degrees of resemblance to the consensus sequence CACN, where N means any DNA base, AGGTGGCAG. That's the consensus sequence. By one way of analogy, one might liken CTCF to a lock that can be operated by multiple keys. So I'm actually reversing the metaphor here. CTF, just look at it as a lock that can be operated by multiple keys. For this reason, CTCF is called multivalent, with multivalence of CTCF zinc fingers modulated by post-translational modifications. It's understandable that one paper describes CTCF as artful. Let me see if I have a picture of this. Hang on. I have the picture of the multivalence. Oh man, no. I want to put a picture of that multivalence. It's just really cool. But anyway, let me go on. In contrast to the multivalence of CTCF, the TFIIIA zinc finger protein can be said to be monovalent, as it binds primarily to only one DNA sequence. So CTCF can bind to multiple slightly different DNA sequences. They're inexact. There can be some variation to the CTCF motif that makes it CTCF a multivalent zinc finger protein. The zinc finger, in contrast to the multivalence of CTCF, the transcription factor 3A zinc finger can be said to be monovalent, as it binds primarily to only one DNA sequence. And the term monovalent is used only informally here, and mostly as a contrast to the term multivalent. But like CTCF, researchers studying it use adjectives of amazement, such as sophisticated, to describe transcription factor 3A's operation. Transcription factor 3A uses an array of 9 zinc finger domains, to bind to an exact 55 base sequence in the promoter of the 5S rRNA gene. However, amazingly, transcription factor 3A uses its zinc finger to bind to the 5S rRNA transcript from the transcript from the gene itself, using a totally different mechanism of nucleotide recognition for the RNA that it did for the DNA promoter in the promoter. Although TF3A is monovalent, I have a lot of spelling corrections I need to make, goodness. Although TF3A is monovalent with respect to DNA, it is polyfunctional because of its dual involvement with RNA. And so I'm going to look here to the side chat. I don't know if you realize how just amazing that is. So the genome has to have 80,000 locations that have the right address sequence. It has to be geographically, physically in the right location on the genome. It has to be like, you know, it has to be oriented like a left or right parentheses. They have to come in pairs of the right, to pairs to be able to form those loops. So that's interesting. And there are 80,000 of these locations that are scattered through the genome. An interesting study would be to see if we had the Sternberg-Collins paradox among primates. I wouldn't be surprised if that were the case. Wouldn't that be cool? Or primates or groups of mammals, because you'd have to be reformatting thousands, tens of thousands of locations simultaneously. That's going to be a real big problem for evolutionary theory to explain patterns like that. And then you have this CTCF protein coordinating all that. I didn't mention that when you saw those loops, somewhere in all of that is the topoisomerase 2 beta protein involved, and we don't know what it does. And you know that that's one of my favorite proteins there. So you have those extrusion loops have some really cool things like the CTCF zinc finger protein plus the topoisomerase 2 beta. Some are involved in there. It's just doing all this cool stuff. And then now we have this. So that's one level of just amazing design. Praise God. The transcription factor 3A is really amazing. So you have a section of DNA in the promoter region of the gene, and it will transcribe what is known as 5S RNA. So this DNA there, you have this stretch of DNA, the zinc finger will attach to it. It'll attach, let me see, it says here 55 bases. That's incredible. An exact sequence of 55. It'll attach to it. That zinc finger then, after, I don't know how this would work, but it would, there's literature on this, and I haven't, it's been a while since I've actually read through it because it's pretty thick. And that's where, the one that's really amazing is where it says, this is a very sophisticated, they use the word, they call this sophisticated. We could say intelligently designed, but they're not going to use that word. It's sophisticated. Sophisticated, it'll bind to that 55 section of DNA. Somewhere along the, in the process of however this gene is expressed, the promoter region will generate this, will convert that DNA into rRNA, which I think is a ribosomal RNA. That zinc finger will then bind to that RNA and shepherded along, I believe, elsewhere in the cell, using a totally different mechanism. Unbelievable. I mean, it's just hard to convey the level of engineering that would be required. I don't think anyone on the planet would know how to build a system that can bind to the DNA and then bind to the RNA that has totally different chemical, not totally different. We know RNA and DNA are similar, but it doesn't mean that that zinc finger can bind to it, not at all, once it's expressed, unless it were designed to. That is very difficult. I don't know that there's any human on the planet, even with all our most sophisticated technologies, that could figure out how to build one like that from scratch, that can do what zinc finger transcription factor 3A can do. And not only that, John Sanford and others published something on DNA that is polyconstrained, meaning if it has like multiple codes to it, you can evolve it in one dimension, but then you end up breaking something else. If you evolve this to make it do a certain function, you can actually break its. So something has multiple functions. Something has multiple functions, and those of you who are involved in computer coding could really appreciate this. If you have a like a Java class that has multiple inheritance, you change the parent class to optimize for one thing. You can kind of break it for kind of the child classes or whatever you call it. I'm not really a Java programmer. You have to be concerned that when you try to optimize this thing to do one task, you can break it in terms of doing another task. So anything that has multiple roles, you have to be very careful how you change things. There was a study done by John Sanford, Robert Marks, George Montañez, and Jorge Fernandez, published in 2011, Biological Information, something. Biological Information, New Perspectives. I happen to have the book somewhere on the shelf there. Oh, and by the way, I got to show you something before we close the stream. You see that top row of books there that I touched? Those are multiple copies of Contested Bones that Dr. Sanford sent me. I have lots of copies to give away, and also of Genetic Entropy. So in this book, Biological Information, New Perspectives, there's something where they said natural selection cannot evolve polyconstrained systems. That is, systems that have multiple use. This transcription factor this transcription factor 3A is a polyconstrained system. It cannot evolve by natural selection as a matter of principle. Just food for thought. Not only do you have waiting time problems, even assuming some natural selection, this thing's going to resist evolution by natural selection. So that, the research on that was in this book. There was a paper that I just mentioned, polyconstrained. So I'm going to survey some of the comments here. Compound cooperation, or lucky. Yeah, I didn't expect this stream to go for 4 hours. I really was hoping I'd be able to get through my talk in 20 minutes. That was not going to happen. But I did want to make a stream where I really had technical talk, because I don't do many of those, you could see, because they're very difficult to conduct. And Patrick, you, Alexander, you, thank you for the kind words. Definitely got my money's worth tonight. Evidence and reasons. Check is in the mail. Laugh out loud. Thank you. Thank you. Yeah, this. I gotta tell you guys. I can't tell you how much I appreciate you guys, because few people will get down to this level of detail. And this is, you know, if an evolutionary biologist, if I were debating, he will just say URVs and science and transposable elements are junk. This would be my response. That less than 1 minute sentence would take like a 4-hour refutation, like I've tried to do here, and say, you got it all wrong, and you don't know how hard this is going to be to evolve that, because all the machinery involved. I showed you these zinc finger proteins, how complex they are, and then also that they bind to these transposable elements. It's so incredible that they could be scattered across the genome to form these extrusion loops. There's not a good evolutionary explanation for why they'd be strategically in the right place to form these loops. And also, remember, they have to be oriented correctly as either a left paren or right parentheses. This is just too much. As Frank Turek said, I don't have enough faith to be an atheist. This is just too much. If people wonder why I'm so passionate about defending intelligent design, you're seeing why. This is declaring the works of God. So, goodness, this has been so fun. And yes, I'm up way too late, and I have I have responsibilities I need to take care of. But I can't tell you how much what a joy it is to share this and to declare the works of the Lord. So, someday I'll try to break this down into smaller discussions. But I think sometimes when you get it all in once, you get just the full impact of the incredible design that God has made for us. So it's hard to say goodbye. May the blessings of the Lord Jesus Christ be with you all today. Tomorrow, I'm going to have Tim Cox join us. I'll be interviewing him. He is running for Virginia delegate for the House, the House Legislature of Virginia, and that's going to be a lot of fun. And this has just been great. So I'll see, Lord willing, some of you tomorrow. The grace of the Lord Jesus Christ be with your spirit. God bless and good night.